You will find a booklet inside your bags. It contains the pictures, the portraits, and the testimonial of different elderly HIV-infected patients around the world that I think that help us to know their needs. So altogether, I think that during these 2 days, during the workshop, we are going to learn a lot to better manage our elderly patients. Finally, I would like to thank BIF and MSD for helping us to fund this workshop for several years. And thank to Maria Sarcopenia from our unit, Fundación de la Lluita contra la SIDA, and Nani from Fundación Huésped in Buenos Aires for helping with the organization, because they did a very good job. So this is the third edition of this workshop here in this building. For me, this is the old San Pau hospital. This was a hospital for many, many years, but the new one is behind this building now. I did my residence here and I love this building. So I think that it represents the best way to get old with dignity, the way that I want my patients get old, age. So we are going to start with some basic talks. After that, we are going to continue with some clinical aspects. And finally, tomorrow, we will finish with some interventions that can help our patients to improve or to age with dignity, as I said. Dr. Clotet is going to start introducing the first speaker, and thank you very much again for coming.
Well, good afternoon. Thank you so much, Eugenia, for the tough work you did to set up everything for this workshop. So it's not easy to arrange such a meeting. And then also I want to thank all the speakers and friends that belongs to the committee, to the scientific committee and faculties. And because thanks to them, we can achieve a good success, which is to debate and to discuss about aging in our patients, something that would have been a dream 20 years ago and now is what we are facing. Then allow me to introduce you the first speaker. It's probably a lecture that you are not expecting to have, but it's something that for me, when I met him in Rome a few months ago, I decided that I would invite him, because at the end, what we have achieved is things that we have never surrendered. And life is a sort of fights until the end, and if you don't surrender, then you live more. What Osman did in his life was exactly the same. He didn't surrender, and then he was capable to leave his country and to reach Europe. And he's going to explain you how tough was that journey, but how good it is for us to learn that many people is still having to struggle and to fight for having a normal life, dignity, and a quality of life that we pursue for all of us, for our patients, but for all of the citizens as well. Okay. Without more words about what Osman is going to lecture, so I want to welcome Osman and to give him the floor, and he will explain us how he did his best to arrive to Europe.
Thank you so much for this opportunity. For me, I think it's a great honor to be able to share my life experience with so many people today here in this specific conference. For me, I think it's very important to make good use of time. I think time is the only thing we can't replace. When my phone gets lost or broke down, I can replace it. Meanwhile, time is something I can't never replace. So I promised to take good advantage of this, your time you've given me, and I promise I will not fail you. So thank you for coming for the first place. My name is Usman Omar, and I'm the founder of the Nasco Feeding Minds, and particularly I'm also the coordinator of Proactivo Pinams. For me, this is a typical poet from Spain called, who said this particular word, not my own word, who said, caminando no hay caminos, hace camino al andar, which simply means that, for example, walking, walking the path is not make, we only make path by walking. So for me, I identify myself a lot with this specific word because this is a summary of what I had to pass through before I could be here today and share this experience with each and every one of you here. The fact is that I think most of us, almost all of us have seen the news from the newspapers, etc., how immigrants, refugees have been struggling every day and night to get to the European continent. But I think it's my debt to explain my life history, to let people know what is really going on deeply there until there's no more histories like mine to explain. I'm a Ghanaian. I was born in Ghana, a very typical village, not more than 100 inhabitants. Fortunately, when I was born, my father was a black magician and my mother died during my birth. In my tradition, in my culture, in my tribe, when this kind of situation happens, the child is considered, something that's supposed not to grow in the culture, in the community. Simply means I'm supposed to be killed because my mom, dad died during my birth, and I killed my mom. So I'm supposed to be killed when I was born. Why am I here? Because my father was a black magician, and he never gave orders to kill me. I was able to survive until 9 years. In my village, we have no access to education, so we have to walk 4 good miles, which is 7 kilometers to go to school in the nearest village. As you can see here, this 2012, when I went back to my village, still, these boys go to school every day, walking 7 kilometers in, 7 kilometers out. Due to the United Nations, for example, the Article 26, all children in the world are supposed to have access to education. As you can see, in a lot of plenty places enough in the world, this particular fact doesn't fulfill its word. Here we have the biggest engineers in the world. I was one of these huge, fantastic engineers. In my village, we don't have access to television, no radio station, no PlayStations. So if I need to play, I have to manufacture my own car, exactly like these boys here. One day, I was just playing with my friends. We saw an airplane fly on the sky. I couldn't understand why an airplane was able to fly and my toy car cannot even move 1 centimeter by itself. I took a stone, I threw it up, it falls over my head. Fortunately, it wasn't so big. If not, I wouldn't be here explaining this story. I started thinking why the airplane was able to sustain. Meanwhile, the stone couldn't sustain on the sky. I was told that flight was manufactured by the white men. And inside that flight, only white people could fly in there. I started thinking that, okay, if this happened, this means that the world doesn't end at the border of my village. No television, no radio. How do you know what is over the borders of your country, your village? Due to this, I was sent to the nearest city to learn street and welding, mechanic. I still have my doctorate degrees grabbed on my hand. I could show you later if somebody wants to verify it. Fortunately, I still have my 2 hands. At the end of that, due to my curiosity, intention to know what is behind the borders of my village, this is the 5-year trekking journey to cross the whole of North Africa, and today to be able to be here and share this experience with you all. When I left Ghana at the age of 13, I felt in the hands of the mafia. When we get to Niger, we was abandoned in this big desert called the Sahara Desert. We was about 46 people. After 21 days, drinking our own urine to survive, only 6 of us was able to make it to Libya. I always said, hell is not up there. Hell is here, depending on which country and where you are, but I don't think hell is upstairs. Hell is down here in the earth. This is really hell. After 4 years living in Libya, the dictator of Muammar al-Gaddafi, I was able to gain $1,800. I felt in the hands of the mafia again. I was told from Tripoli it would take us only 45 minutes to go to the paradise, which is Europe. 45 minutes. from Tripoli. Illiterate, no access to information, no access to education, I felt in the trap of the mafia again. I gave all this money to the mafia. We left Tripoli, Tunisia, Algeria, Morocco's, Western Sahara, Mauritania. It took us almost 3 months to get to Mauritania. Once we get there, the boat, we have to manufacture the boat ourselves. We was given wood to make it ourselves. As I said before, I'm a mechanic. Welding, I never work with wood. This is a real example that almost 95% of people who have been intending to cross to Europe sink and they are under the Mediterranean Sea. This is the reality that no newspaper will show to you. For example, this picture is real. That was last year. However, due to the Syrian crisis, the mafia have changed their strategy. But as you can imagine, 2 days, 48 good hours, sitting on this boat in a sea, you don't know where you are going without food. Definitely, as I said before, I think hell is down here in the earth. In my case, after these 2 days, we was able to make it to the Fuerteventura Island. Most of my friends died on the way. Musa died on the first attempt to cross the sea, and hundreds of them more. After a couple of 1.5 months in the prison, I was sent to Barcelona because I was a minor, and I had the permission to stay in Europe, in Barcelona, in Spain, in Gerard. I was asked which part of Spain do I will to stay. I had no idea of Spain. I knew only Barca, which is FC Barcelona, due to the football match I watched the first time I had the opportunity to watch TV. On the 24th of February, I was sent to the city of Barcelona, alone. I was 17 years, illiterate, no Spanish, no Catalan, no friends, no telephone. For me, this was the huge and the worst jungle I've ever been. It was February, very cold, very cold. Anyway, I stayed in the street for almost a month. Fortunately, I met this woman. I just asked her I wanted to go to the Red Cross. That was the only thing I knew about. That was the only thing I was told in the prison, that when I get to Barcelona to ask of. She didn't speak English, but she wanted to understand what I was trying to explain to her. So she took my hand, we stand by the roadside, she took off her phone and called her husband and gave me the phone to explain to the husband what I needed from her. He asked me hundreds of questions. But I still remember when he asked me how old I was. I told him I was born on Tuesday. He said, "Oh, fantastic, but how old are you?" I said, "Oh, what do you mean? I've already told you I was born on Tuesday. What else do you want?" In my culture, the most important is the day of the week you are born. It doesn't matter how old are you, either you're 23, 50, 60, it doesn't mind. For example, Kofi Annan used to be the US Secretary General. Kofi Annan is a Ghanaian. Kofi means he was born on Friday in Ashanti. So in my culture, this is the reality. However, he didn't give up. After a couple of days, I was sent to the Red Cross together with a woman, and they became my adopted family. Here, I became a born again. I had opportunity to learn Spanish and Catalan, read and write. I started my high school. I studied chemistry 2 years in the University of Barcelona because I wanted to understand whether the black magic was real or science was really giving the solution to sickness. After 2 years, I quit from chemistry and I studied public relations and marketing. I graduated from a degree last year and I did my master's degree in a study about managing of social initiatives. For me, after having this access to this opportunity, I thought the best I could do is to share my luck to other people. When I get to Spain, this is my younger brother. His will was to sell all the animals we had there to follow my footsteps. It wasn't easy to convince him that the solution is in Africa, in his country. He only needed to go to school. But at the end of it, I was able to convince him. I paid for his education in Ghana. He studied political science. At the beginning of this year, he is inaugurating his own startup company. He has 8 people working with him right now. More than 62,000 people have been registered in his platform. The solution is feeding minds, giving them the right tools. They would save themselves. So together with him, we thought we should do something else. I've saved his life, but there's hundreds, millions of them. That's how, together, we established the Nasco Feeding Minds NGO, 2012. Basically, what we do is we facilitate ICT centers for schools so they could learn today. Digital education, for example, is the most powerful to get access to information any corner from anywhere you are in this modern world. This is our first lab in 2012. I had to buy the first 45 computers with my own pocket money because nobody believed on what we was proposing. When I see this picture, it still makes me, I feel affected emotionally because you couldn't imagine how I struggled to get this first 45 computers for 850 students in the north of Ghana. We hired 2 teachers, and that's how we started our first computer lab. Normally, they said, if you trust in your effort, your belief, you always have a reward. This was last 2017. We was rewarded by the International Telecommunication Union in Geneva, Switzerland, and a specific organization from the United Nations. And of course, I invited him to come to receive the award together with me. When he comes here, he didn't come with a small boat. In 5 hours, he was in Switzerland with me. It took me 5 good years to get to Europe. They can come in a different way. Our initiative attracted a big NGO called Proactive Open Arms. It is a specific and the most strongest NGO rescuing refugees and migrants in the Mediterranean Sea since the last 3 years. Together, we think the solution, of course, is in the source root of where these immigrants come from, not in the Mediterranean Sea. So we established this new collaboration, which means we need to feeding minds, saving lives. And that's what we need to do, going back to the source root of the people of where these migrants come from. Of course, the solution is not here. I understand when in case of urgencies, for example, we need to do something. Like these cases, we need to do something. But the long-term solution is in the source root. I will show a short video of what the work of Proactive Open Arms. It's quite tough, but I think it's necessary for you to at least make an idea what exactly it is. This is a normal day of Proactive Open Arms. Sorry. This is a normal day of Open Arms in a sea. I hope you remember when you saw the boat for the first time, it was full, and you couldn't see what was under the boat. Now let's see what's under it. This is the reality. that you will not see from any TV stations. This is what Proactive Open Arms sees every day they go to rescue. I hope nobody will to see this in his work every day. We need to take action. If not, any moment, any day, we will swim in our beaches, our wonderful beaches, with dead bodies. I think each and every one of us, we need to take action. So, what have we done? What have we achieved during these 6 years? We've established 5 ICT centers. Since 2012, more than 10 schools uses these ICT centers. Since then, we've educated more than 11,000 students in 6 years. We've also given beekeeping education to 24 women who are now making honey in the north of Ghana, and more other parallel projects we've also done. Why do we need your support, or why do we think this is the most important? And of course, you need to support us to continue. Basically, to maintain this structure, to maintain teachers in all centers, it costs us 420 euros a month, which is about 5,000 euros a year. Of course, without support, it's impossible to continue maintaining this structure. And of course, I think all of you agree that the solution is to give them information and access to education, just like I did with my brother. So, of course, we need to continue. And to continue, of course, we need some economic support to be able to sustain this project. Well, we also think all of you can, if you want to support, you don't know how, you can become a partner, or you can make a direct donation in our website, or on our Facebook pages, etc., etc. And of course, after the presentation, I think Dr. Clotet will maintain our presentation in his website, so you can get more information about it. And so, thank you very much. I hope I didn't misuse your time. Welcome.
Thank you, Osman, for sharing with us this experience. I don't know the word to define your story. I suppose it's incredible, or I don't know, my English don't, I need more adjectives to define this story. I think that I have to learn a lot from you. Thank you very much. We are going to start with the first session. This is the most basic session that includes talks about cellular senescence, immunosenescence, and inflammation, and also microbiome and aging. And we have three speakers, experts in these fields. The first one is Dr. Marta Martín from the Universitat Autònoma de Barcelona, from here. And she's going to talk about DNA repairment and cellular senescence. The second one is Julia Blanco. I prefer to introduce all of you. And no, don't worry. The second one is Julia Blanco from Irsi Caixa Foundation, from here, my center in Hospital Germans Trias i Pujol. He's going to talk about immunosenescence and inflammation. And the last one of this session, Dr. Sergio Serrano from Madrid, who is going to talk about microbiome and aging. If you want, I think that it's better to have the discussion at the end of the three. Okay, thank you.
So, good afternoon, everybody. I hope my talk won't be too basic, but in any case, I hope you enjoy it. My name is Marta, and I work at Universitat Autònoma de Barcelona. And I'm going to talk about the contribution of DNA damage to aging and senescence. Aging is characterized by progressive loss of physiological integrity that leads to impaired function. And ends in common diseases like diabetes or heart failure, these kind of diseases you know better than me. All mammals age and most organisms age, but it's difficult to define how aging begins, or because it's very multifactorial. So I follow this paper, very nice, in which they describe 9 hallmarks of aging. We are not talking about all of them, but all these hallmarks are present in aging cells, aging tissues, and aging organisms. And when you experimentally alleviate them or aggravate them, the aging process changes. It gets ameliorated or it gets worse. So I'm going to focus on the hallmarks that are more related with DNA damage, because DNA is the central molecule of life, and it's been described everywhere that with aging, a lot of DNA damage is accumulated in cells. So there's a lot of exogenous or endogenous damage that can damage the DNA. So we generate, for example, oxygen species, but also external damages like radiation or chemicals. They can damage DNA, and they can generate multiple kinds of lesions. But the lesions more important for us, or more deleterious for DNA, are double-strand breaks, which are here, in which both strands of DNA are broken. These are really dangerous, because if we get a lot of double-strand breaks and they don't rejoin like they were originally, we can get mutations that can, in the end, be potentially oncogenic. So the cell gets, every cell in your organism gets between 10 and 50 double-strand breaks every day. So this is a lot. Unfortunately, fortunately, cells have a lot of repair processes to cope with all these breaks. What the cell does is to start the DNA damage repair pathway, or the DNA damage response, in which, first of all, stops the cell from progressing, from dividing. The cell can't stop in any of the phases of the cell cycle, and then a lot of DNA repair proteins are recruited to the DSB site to be repaired. There's a lot of, but there's many repair pathways. These are the both most important repair pathways of double-strand breaks, which are the non-homologous end joining and the homologous recombination pathway. The most important difference is the non-homologous end joining pathway is able to function during the whole cell cycle. And this is why important, because a lot of cells do not divide anymore while we are alive. For example, neurons, they are always in G1, so they only have one repair pathway to rely on. The other pathway, the homologous rejoining pathway, is restricted to S and G2 phases, which means after DNA has replicated. So it can only work in those cells that replicate, that actively divide. Also, homologous rejoining is more faithful repair pathway, because this pathway uses the template of the other homologue, of the chromosome that's homologue to the one that's broken, to be repaired. So repair is always, always faithful. With non-homologous end joining, what this pathway does is to very fastly rejoin the ends of 2 broken chromosomes. But if there's too many broken chromosomes, or if the repair gets a little bit slower, mistakes can arise. So what happens when we have these breaks in the DNA of our cells? First thing can happen, and we all expect, is that the repair is correct, the cell goes on, cycles again. Another thing that can happen is the damage is too big and the cell cannot cope with it. So one thing the cell can do is to die by apoptosis, or it can stop replicating and enter in a senescent state. This cell is metabolically active, but cannot replicate anymore, it cannot divide. So it cannot transmit the damage that's carrying with her. What we don't like, but sometimes happen, is that repair is incorrect. If we accumulate incorrect repair, then we can activate oncogenes, or we can suppress tumor suppressor genes. And this is a common finding in aging, and also in cancer, which is also a disease related with aging. So, in age cells of mice, rats, but also human, we find accumulation of breaks which are not repaired, or breaks which have been incorrectly repaired. Also, another evidence that links aging with double-strand break repair or with repair efficiency is that those individuals who have mutations that affect DNA repair proteins present a premature aging phenotype. These phenotypes do not phenocopy all characteristics of aging, but quite a lot of them. So there's another evidence of how much DNA repair or maintenance of genomic stability is a must for the aging process. The big question here is, do cells accumulate more damage just because we've been spending a lot of years alive, or it's because the repair mechanisms function less efficiently? This is something that we try to answer, a lot of research groups try to answer, and something my research group tries to do, and if I have time, I will explain later something about it. Another hallmark of aging is telomere shortening. You know, telomeres are stretches of DNA at the end of chromosomes, and these ends of chromosomes, they form a loop on themselves to avoid the repair machinery to fuse them. And here you can see chromosomes with the 4 telomeres that we should have. It's been described that with age, you can see up on your right, on my right, sorry, how telomeres shorten with every cell division in somatic cells. This is so for everybody. The only cells that do not lose telomere length are stem cells, germ cells. But all the other cells, with each cell division, we get shorter and shorter telomeres. When telomeres get too short, they also activate the DNA damage response, because if not, these ends, which have no telomeres, they can get reorganizations between them and function as dicentrics that in the end will break during mitosis, and we have 2 cells with more unrepaired breaks that can combine between them, and this is the onset of genomic instability. So short telomeres are very efficient at starting the DNA damage response, halting the cell, inducing senescence. And it's been described that mice with short telomeres have a decreased lifespan. Mice with longer telomeres have a longer lifespan, so telomeres definitely influence aging. This very nice experiment in which adult animals, in which telomeres had begun to shorten, were experimentally transfected with the telomerase enzyme, which is an enzyme that is able to elongate telomeres, and this elongated the life of these mice. So finally, the last reference I show you is a very new work in which they found a very strong correlation between short telomeres and mortality risk, and this they have found in young people. So it means maybe aging can be predicted. I don't know. I just throw here the question, but maybe checking the telomeres at a young age might be a good idea. This is something we can leave to discuss. Another hallmark of aging is epigenetic changes. All these are epigenetic changes we can find in all cells as they age. For example, methylation of the DNA changes, and this affects basically transcription of genes, which are tumor suppressor genes, and this transcription is diminished, so the neoplastic transformation is favored. Also, there are less histones in cells which are aged, so that means that we have a more relaxed DNA damage, and it's more prone to be broken, and gene expression also change. One of the stones especially related to DNA damage is the one up, H4K16, is the H4 histone, which is acetylated in lysine 16. And this histone is very, very strongly related to DNA repairability. It diminishes with age, although there's a lot of contradictory results. It's now beginning to be clear that it's reduced with age, and it has a role in recruiting DNA repair proteins. So its reduction with age implies that DNA repair will be less efficient in these cells, so more breaks will accumulate. So everything is the same. Finally, also, changes in heterochromatin are linked with aging. When we age, heterochromatin diminishes a little, but most of all, it gets redistributed. If a gene is compacted in an heterochromatin structure, it's almost impossible to be transcribed, and all these changes in transcription, in DNA damage, and in chromosomal instability drive aging. And also they drive cancer. Lately, a lot of effort is done at the research level, but also at the pharmaceutical level with the sirtuins family. These are deacetylases, so they are proteins that deacetylate histones, and are very good targets for aging. It's been described that mostly sirtuin 6, loss of function, reduces longevity. So its gain of function extends longevity in mice. It has not been proven in human yet, but they are trying to settle the experimental background to prove so. These sirtuins modify the histones I was telling you before, but also are related with DNA damage. So this is a new, also a very active, how do you say, research area now relating to aging. I don't know many things about mitochondria, just the things I have to know being a teacher, but this I found very interesting because when we think about mitochondria and aging, we always think of oxygen species which are toxic for the cell because they generate oxidation. But lately, it's being increasingly clear that mitochondrial DNA accumulates a lot of mutations, and that these mutations are also involved in aging. I found this paper that might be interesting for you, in which they found how mitochondrial aging was accelerated by this antiretroviral therapy. But why? Just because it favored the accumulation, you can see here, the ones treated with the antiretroviral, they had a significant higher number of mitochondrial mutations. So these cells have a mitochondrial dysfunction. If mitochondria does not work well, these cells have less energy and they do not function well. So it favors the dysfunction of the affected tissue. These implication of mitochondrial DNA in aging is quite new, and it's becoming more and more important because more and more works are being done. Nobody was looking at this, and it's being a new window of research that we are liking very much. I don't know if you know something about reactive oxygen species. I'm sure you know, but for me it was unexpected because recent reports are saying that maybe ROS were not so bad as we thought, and low levels of ROS might be even good to boost in the cell stress response that would help this cell to cope with ROS and to even extend lifespan. This would be only true if ROS accumulation would be not so big, just at the beginning of the mitochondrial dysfunction and at the beginning of the accumulation of ROS. But I find this very, very interesting because now that we are more able in the lab to measure the quantity of oxygen species, it's when we are seeing that maybe they do not contribute so much to mitochondrial damage nor to mitochondrial dysfunction as we thought. We have to study them more, but maybe we should focus more on mitochondrial DNA alterations. In aging tissues, we always find senescent cells. Cellular senescence means a cell which has stopped dividing. It does not divide anymore. And it does not divide anymore because it carries a potentially oncogenic stress. To say so, it carries a lot of DNA damage that cannot repair, or some oncogen is being activated. So the cell senses this potentially oncogenic stress and stops dividing. The cell is active, is metabolically active, but it does not divide anymore, so it does not spread this DNA damage to the progeny. Organismal aging, sometimes it's called cellular senescence, but for me it's not exactly the same because senescence is a cell phenotype. So what we see is that with age, the number of senescent cells in tissues doubles with respect to normal age. We have to understand that this process is natural. Some cells, when they accumulate a lot of damage, they die, enter apoptosis and die. For example, leukocytes. But some other cell types, when they accumulate a lot of damage and they cannot repair, they enter senescence. For example, fibroblasts or epithelial cells. It depends on the cell type. So this is a normal mechanism to cope with DNA damage, but as we age, more and more senescent cells are found in tissues from all people. So these cells stop dividing, they change shape, they get ugly, as we say in the lab, and this is caused because of telomere shortening, accumulation of DNA damage, activation of tumor suppressors, basically the same hallmarks of aging. Senescence is activated by the common pathway, or has a common pathway with the DDR, the DNA damage response, which is the p53 protein. When a cell enters senescence, depending on which of these factors generate senescence, is activated by p53 or by p16. So this is important. I tell you this only for one reason, is because maybe when we want to check if a tissue is old enough, we would like to see how many senescent cells are in there. And one way to look at it is to check for p16 or p53 levels by Western blot, for example. Another way to look for how many senescent cells do I have is to perform a test if these cells divide or do not divide anymore, by checking Ki67 or doing the thymidine incorporation test. Also beta-gal, which you can see on the right, it's a very common a technique that we use to detect senescent cells. All the blue cells you can see are senescent. Senescent cells accumulate this foci of heterochromatin. You can see here, this foci. And also, they accumulate a lot of DNA repair proteins, and they accumulate all together in places where DNA repair cannot be performed. And these are called DNA scars, which are accumulation of a lot of DNA repair proteins. And these, that we call foci, that can be seen in the microscope by immunofluorescence, all these foci can persist for days or months. And this is another characteristic of senescent cells. Because they do not divide, They arrest because they cannot repair, but because they cannot divide, they don't expand this damage. Mm-hmm. Finally, another characteristic of senescent cells is that they have a special secretory phenotype. They secrete a lot of cytokines, growth factors, and metalloproteases. So they generate inflammation and an immune response. What we think is that this is the strategy of the cell. When an organism is young and a cell gets damaged, and this cell undergoes senescence, it's a way to call the immune system to get rid of this cell via this secretory phenotype. But what happens when the cell gets old, when the organism gets old? We find that we have an accumulation of senescent cells. What happens here? Do cells have more damage? Or maybe the immunological system cannot get rid of all the senescent cells because it does not function like before. Or maybe everything is happening together. These are things that we still do not know. But we know is that in aging, there are more senescent cells. The secretory phenotype, then it's more relevant. So there's more pathologies associated with this secretory and inflammatory phenotype in the places, in the tissues where there are a lot of senescent cells. For example, it has been just 2 weeks ago published that these researchers have found a new drug that is able to clear senescent cells from glial cells, and so to prevent tau aggregation, which favors Alzheimer, as you know. And the only thing that this drug does is to get rid of senescent cells. So all this secretory phenotype is reduced, and it alleviates these tau depositions. So there's a lot of research to do here, and these are many pathologies in which we find an increased frequency of senescent cells, which are a lot of them, and all of them are aging diseases. What we don't know, of course, these are hallmarks. We don't know if they are the cause or the consequence of aging. This is the big question here. So this has been a small overview about aging and the hallmarks of aging. And now, if you allow me, I just want to explain a little bit more about DNA repair, of a bit of what we do. We want to try to know if accumulation of DNA damage in cells, it's because simply we accumulate damage throughout our life. So when we are old, we have more unrepaired DSBs in our cells, or because the DDR does not function so well. So we work with cells from the mammary gland of women below 27 or older than 60 years old. We get these cells, we culture them in the lab, and we irradiate them, and we analyze how many breaks remain after irradiation. And this is a way to evaluate the efficiency of the repair. How do we do it? We count these foci, which are marked by a phosphorylated histone. This is an universal mark of breakage. When the DNA breaks, the H2 histone gets phosphorylated. So if we detect this, it's a good surrogate of double-strand breaks. Every foci means there it has been a break, so we can count. And this is what we do. We count on the microscope. It's a hard work, I can tell you. And what we found is that before irradiation, you can see that there are the number of breaks. All donors, they always have more breaks than young donors in their cells. And after irradiation, it's the same. It does not matter which time we evaluate, first hour, second hour, 1 day after irradiation, cells from all donors always have more breaks. We have evaluated the speed of repair, and you can see that the speed is the same for all donors, which is the red line, compared to young donors. The problem is at the beginning. All donors seem to start to repair later, a little bit later. And this might favor the accumulation of some breaks that do not find a partner to repair. What we are just finishing now is we induce local damage so that we can differentiate this damage from the damage that was there before, because I've told you that all cells have more damage, so I want to be able to differentiate new damage from old damage. And we count how many repair proteins I can find in any break. We look for 53BP1 and BRCA1, which are, it doesn't matter the name, but they are proteins from the homologous recombination or the non-homologous end joining pathway. We are now finishing to evaluate these, and we are just finishing, and we want to publish 2 papers about this. It's about to be done, but we found that 53BP1 recruitment is delayed in cells from all women. So it means there's a repair deficiency, at least in these cells, linked to age. This is what we think, our proposal. Another thing we do is analyze epigenetics, and the epigenetic mark we analyze is H4K16 acetylation, which has been linked with 53BP1 recruitment. And this mark, this is the DNA, and it's wrapped around the nucleosomes, and in here, the histone 4, is acetylated by MOF, and under these circumstances, it can recruit 53BP1. The sirtuins, which I told you before when I talked about epigenetics, are the ones in charge of eliminating these acetylation signals. The equilibrium between acetylations and deacetylators in the genome are responsible for maintaining the heterochromatin and euchromatin in the right proportions. So DNA then is properly relaxed or is more condensed, so it's more accessible to DNA repair proteins or not. What we found in senescent cells, which are not age cells, remember, now we know this, and also in cells that we've aged in vitro, so we've been accumulating passages in vitro, like 20, 25 passages, so trying to mimic organismal aging, which is not possible, but it's similar. These in vitro age cells are not senescent, they divide, but they accumulate a lot of divisions. Both of them, they have low levels of H4K16, meaning this is altered with this kind of aging processes, and this we've checked with many techniques. And you can see that here in this graph, what I show you is cells at different passages, 5 passages in culture, 10 passages in culture, 25 until 30 passages in culture. When cells are kept in culture, 53BP1 recruitment it does not function as well as in young cells. And this we can see here again. In old cells, you can see less 53BP1 than gamma H2AX. And in these cells, also you can see a reduction in H4K16 acetylation. So we think there's a relation between these two hallmarks, and what we think is that loss of H4K16 acetylation limits recruitment of 53BP1. When we induce DNA damage, this phenotype is reverted because cell is stressed and the DDR is activated. Some acetylators are recruited, HK416 is recovered, and 53BP1 colocalization is also rescued. So we are presenting this model in which we think HK416 acetylation must be kept under certain levels in order to ensure a correct DDR activation. If HK16 acetylation is lower, then the DDR does not function properly, or DNA repair proteins are not recruited fast enough to the DSBS sites. And just to end, I want to show you my group, because in research, everything has to be done in a group, and nothing is possible alone. And Lourdes and Teresa, which are the two girls in the back, are the ones who have done most of the work, and I'm thankful for that. And hope you've enjoyed it and learned something. I'm very thankful for being invited to be here. Thank you very much.
Okay. Doesn't work, the pointer? No. No. Oh, there's the one. Okay, so let's move from the general concept of senescence to the more specific concept of immunosenescence and also to inflammation. This is my conflict of interest. Both inflammation and immunosenescence are natural processes. And all of you know that the inflammation can be widely defined as a natural immune response to danger. The danger could be a pathogen, it could be physical damage, and this inflammation is mediated by a lot of molecules that will recruit lymphocytes to the site of inflammation and a large collection of molecules that will mediate the tissue repair. The immunosenescence, which is also a natural process, is the aging of the immune system and is associated to a loss of function of our immune system at older ages. And this is clearly illustrated. I always put this slide in my presentations because I think that this is very, very clear. If you follow the black dotted line, this is the number of infectious diseases related to hospitalization in men, and you can see a progressive increase over starting at 60, 65 years. And this is associated to a lack of function of the immune system and to an accumulation of memory T cells. To view a little bit more in detail what is happening when we age in our immune system, you can follow first the black line. This is the number of thymic emigrants. The thymic emigrants are naive cells, which are the younger cells of in periphery in our immune system. And you can see that the number of new cells is decreasing with age. However, the total number of lymphocytes is maintained in a stable number. This is the homeostatic control of our immune system. This control is mediated by the duplication of the cells. So, if you look at the red line, you can see that this duplication of the cells, as Marta told us, is associated with a loss of the telomere length of these cells. And then, when we get 65, the number of naive cells decreases because the cells are no longer able to duplicate and they enter in a more senescent state. Okay, what happens in HIV infection? All of you know what happens in HIV infection. There is much more inflammation than in HIV and infected subjects. And why is this inflammation increased? But obviously by the presence of the HIV, the virus is a pathogen, it will mediate an inflammatory response. But there are several specific features or characteristics of the HIV infection that will worsen the inflammatory status. The first one is that HIV is infecting cells from the immune system, so is limiting the capacity of the immune system to respond to pathogens. And the second situation that will increase the inflammation is the destruction of the gall, the gut-associated lymphoid tissue. And this occurs very early in the course of the infection. During the primary infection, there is a huge destruction of the gall tissue. And this has specific implications on inflammation. Let's see. The gall contains most of our CD4 T cells because they are there to control all the bacteria that are in our gut. So there is a huge number of lymphocytes that are organized in lymphoid tissue into the gut. HIV destroys these cells and destroys the structure of the lymphoid tissue. And you can see here the staining of CD4 T cells in a healthy or in a, on the right, in a HIV-infected gall tissue. This destruction of CD4 T cells will lead to a loss of immune function at the epithelial barrier. So the bacteria that are in the lumen of the gut will cross the epithelial barrier, and we can find them in our systemic circulation. And this is what we call bacterial translocation. And we can measure that using LPS, which is a product of the bacterial wall, or measuring the presence of bacterial DNA in our plasma. In both cases, you can see that HIV-infected individuals in the chronic phase of the infection, they show higher levels of LPS or higher levels of bacterial DNA than HIV-uninfected individuals or very, very early HIV-infected individuals. Why these bacterial products are important? Because they are able to activate immune cells, because they bind to the TLR receptors that are expressed on our immune cells. This is the receptors that detect the presence of bacteria in our body. One of the consequences of the binding of LPS, for instance, to monocytes is that it may increase the differentiation of these monocytes, and they will lose CD14 on the surface. They will increase the expression of CD16, and this is something that we can see here, and they will produce inflammatory cytokines such as TNF. So here you can see that HIV-infected individuals shows in periphery a higher percentage of CD. Okay. Thank you. Okay, so perfect. So you can see here how the HIV-infected individuals show higher percentages of activated monocytes. Which are the consequences of that? I think that there are two, maybe, let's say, three consequences. The first consequence is that activated monocytes are more prone to associate with plague, so they are associated with cardiovascular disease, and they also may translocate to the central nervous system. So this activation of monocyte could be associated with neurodegenerative diseases in HIV-infected individuals because of the presence of these inflammatory signals associated to macrophages and monocytes. But the third consequence is that, as you know, the immune system is not a collection of isolated cells. All the cells in our immune system are connected, and they talk through cytokines. So the higher production of cytokines from monocytes will also activate other immune cells. And specifically, T cells are relevant for that because the activation status of T cells will control the replication of HIV. So activated cells are able to replicate very easily HIV, while quiescent CD4 T cells are not able to replicate the virus. So this led to a, let's say, a first inflammation circle in which HIV destroys gut tissue, produces microbial translocation, immune activation, a general immune activation that will boost HIV replication, worsen the destruction of the cells in the gut. So this is a vicious circle. What happens during HART when we are able to control viral replication through therapy? I think that there are three main aspects that are relevant. The first one is the persistence of the virus. We control viral replication, but there are still viral antigens in the body. The second relevant aspect is the incomplete tissue repair. So not all treated HIV-infected individuals are able to recover normal levels of CD4 T cells. And the second one is the immunosenescence. During untreated HIV infection, the immune system has been working very, very hard to control HIV replication. And this has a price. I mean, the telomeres have been shortened, and the number of duplication of our lymphocytes has been higher than in HIV-uninfected individual. And the price is an accelerated immunosenescence. Let's see a little bit more in detail what happens in treated HIV infection. I think this is a very, very classical slide. This is showing the activation of CD4 T cells in HIV uninfected, treated, or untreated individuals. And this difference is also applies for any or most of the inflammatory markers. Treated individuals show higher levels of immune activation than healthy individuals. Obviously, ART is able to reduce activation, but it doesn't normalize the levels of activation. And the same happens for almost all the markers of inflammation. So there is a residual inflammation in our treated patients. How can we minimize this inflammation? Obviously, optimizing treatment. This is a recent paper from Montse Plana at the Clinic Hospital in Barcelona. She shows that, for instance, in monotherapy with PIs, some inflammatory markers are higher than in standard antiretroviral therapy. So we have to be sure that we are controlling completely viral replication to reduce the level of inflammation in treated patients. The other relevant parameter for inflammation is the level of CD4 T cells. These are data from several inflammatory markers and the association with different parameters of these patients, and in particular CD4. Most of these inflammatory markers show a direct and negative association with the number of CD4 T cells. So a better recovery of CD4 T cells will reduce the level of inflammation. But there are other factors that are relevant for our inflammatory status and our immunosenescent status. And one of them are the pathogens that co-infect HIV-infected individuals and even HIV-uninfected individuals. Most of our lymphocytes, especially those that are in tissues, are specific against chronic viruses. The most relevant one probably is CMV, but also EBV, and we can find in the spleen a lot of memory cells against EBV, but other recurrent or chronic infections. So these infections may also control or regulate the status, the inflammatory or immunosenescent status of our cells. In the case of CMV, this is our work also from the lab. We measured, Cecilia Cabrera in that case, measured the reactivation of CMV in different HIV-treated individuals, and she found a higher level of CMV reactivation in patients with low CD4 T cells. So this is telling us that there are subclinical reactivations of CMV in hard-suppressed individuals. And this is relevant. This is relevant for the senescence of our T cells. Because, as I told you, when we are young, we have a lot of naive cells, but as Marta told us, those cells divide, they differentiate, they lose telomeres, and they get a senescent state. Most of them will die, but some of them will get to this senescent state. And the speed at which naive cells travel for this differentiation status is reflected by the immune activation. And immune activation is controlled by inflammation and also by pathogens, specifically CMV or HIV, obviously, another chronic virus, but also, and I hope that Sergio will go in detail about the microbiome, but also by other pathogens that are in the microbiome. All the bacteria that are in the microbiome may control our immune responses. How can we measure the level of immunosenescence in our immune cells? We can measure the percentage of T cells in those stages, in naive, central memory, transitional memory, effector memory, or senescent stages, using different markers on the surface of those cells. Terminally differentiated or senescent cells epigenetically downregulate CD28 and CD27. These are activation molecules, so downregulating them, senescent cells are more difficult to activate and to divide. And also, these senescent cells will express CD55 molecule. This is usually a marker of replicative senescence of our lymphocytes. And obviously, we can also measure the telomeres in our cells, and a decreased telomeres is also a feature of senescence cells, as in general the senescence. This is just an example on how CMV may control senescence of our immune system, and this is a direct comparison of individual CMV negative or CMV positive individuals and a measure of the number of CD8 cells or the number of CD8 cells that lack CD28 expression. Those cells, those terminally differentiated cells, are higher in CMV. And also, is relevant, the infection by cytomegalovirus, CMV, is associated with increased mortality in older populations. So this is also true for HIV. In fact, the immunosenescence status in HIV is dependent on CMV serostatus, but also on the level of CD4 in our patients. Higher age, higher CD57, lower CD4 cells, higher CD57 expression in our immune cells. Can we calculate how older is the immune system in HIV-infected individuals compared to HIV-uninfected? So this is very difficult, but there is a paper, a very interesting paper using very complex analysis of immunosenescence features in HIV-infected individuals that quantifies in 4.9 years the difference in immune age between HIV-infected individuals and HIV-uninfected. Which is the impact of immunosenescence in the clinics? There are few information about that, but I think that at least there are two main impacts. One is on the CD4 T cell recovery. And this is a paper from Jose Ramon Blanco, this is not me, in which they show that the length of telomeres in HIV-infected individuals, especially in older ones, may determine the recovery of CD40 cells after HART. And also inflammation is associated with this recovery. This is a paper from Yolanda Pacheco showing that higher levels of IL-6 or higher levels of CRP are associated with lower immune recovery. So more immunosenescence, more inflammation, less immune recovery. And what happens after HART? So the levels of activation and inflammation, and immunosenescence are also associated with carotid lesions in HIV-infected individuals. In fact, those patients with these lesions show higher levels of immunosenescence or higher levels of activation than HIV-infected individuals without this kind of lesions. So this makes the scenario a little bit more complex, and we can add a second circle to the first one. In that second circle, we have to think about immunodeficiency, about the number of CD40 cells in our patients that will determine the inflammatory and immunosenescence status, and will also help to reactivate chronic viral infections. And those chronic viral infections will increase the immune activation in our patients. And finally, this immune activation will accelerate the process of immunosenescence that is associated with the morbidity and mortality of our patients. What we can do to revert this effect, we can try to reduce the activation, the immune activation or the inflammation in HIV-treated patients. And this has been done with different approaches. Peter Hunt and Steve Dix add valganciclovir to HIV-treated individuals to block CMV replication, and he got a partial decrease of immune activation. Others have tried anti-LPS antibodies with no effects on activation. Chloroquine, which is, let's say, has some immunosuppressor activity, may reduce CD8 T cell activation, as COX-2 inhibitors also. Statins are also immunomodulators, so can be also added to HART to try to reduce inflammation. And finally, probiotics, and this is something that Sergio will touch in his talk. But none of those approaches have yielded good enough results, I think. So there is no clear option right now to reduce activation. Or we really need more clear data on the effect of these interventions, on the clinical effect of these interventions on survival, comorbidities, etc., etc. So what we can do right now, optimize therapies probably, and treat as early as we can. This is probably the best approach to reduce activation and inflammation in HIV-infected patients. When we talk about treatment, obviously we have to block HIV replication to reduce inflammation. But there is a recent paper that suggests that HART composition may impact the length of telomeres in our immune system. The mechanism is not clear, but in the context of the NEED-01 study, in which there is a comparison of raltegravir with tenofovir FTC, José Ramón Arribas measured the length of the telomeres in PBMCs from those patients, and this is at the baseline, and this is at week 96. In general, there is an increase in the length of the telomeres, but the increase seems to be only associated to the tenofovir FTC branch, no in the raltegravir arm. This is not clear how this may lead to a clinical benefit, how these, which are the mechanisms that are controlling this effect. But it's at least interesting result. The second approach is the early treatment. The early treatment doesn't block the increased inflammation, but it will reduce the impact of inflammation, activation, and senescence on the immune system. The most clear impact is when treating individuals. These are two papers from Gintanat, treating very early infected patients. And the most clear effect is when treating patients in FIBIC1. Obviously, this is very, very difficult to do it in the regular clinical practice. So just to finish, I think that the main take-home message is that immunosenescence is a progressive process boosted by inflammation and immune activation, and occurring mostly in untreated HIV replication. It's relevant to think that other infections, such as CMV, may contribute to both the immunosenescence and the inflammatory status of HIV-infected individuals, and that both immunosenescence and inflammation may limit the recovery of CD4 T cell counts and may impact non-AIDS-related comorbidities. How to block that? This is a very, very difficult question. We have very few and unclear therapeutic approaches to limit or to reduce this immunosenescence or inflammation. And thank you very much for your attention.
Okay. Hi, thank you, Eugenia and Ventura, for the kind invitation. It's fantastic to be in this wonderful city, beautiful hospital, in this very nice audience. So I've been asked to try to reconcile the microbiome studies in HIV with the pathogenesis of aging, inflammation, in the context of HIV-infected patients. And it's going to be frustrating for me to summarize the evidence because there are plenty of beautiful studies. And it's always a good exercise in the microbiome presentations to step back and get rid of our classical training in infectious diseases linking microorganisms with diseases. And it's probably a good idea to imagine ourselves as ecosystems because indeed we are the trillions of microbes that inhabit ourselves are so complex as any other ecosystem. And for example, here you have a grass close to Portugal, Cáceres, in Spain, and this grass is mainly formed by this leguminous, which is very good for shepherding, and it forms a very stable ecosystem, resilient to perturbations, and perennial. It doesn't change over the year. And if you look here, you see that there's a huge change. Okay, here because here this grass was digged 3 years ago, and this grass changed into this other ecosystem formed by a multitude of flowers. Which are opportunistic. They die every summer. They change. It's a very unstable ecosystem, and actually it's also very resilient because it's very hard to restore the ecosystem to the previous state. And these changes in floral ecosystems following perturbations can even be more dramatic. And here you have this plant. It's called Viborera in Spanish. I don't know in English. Sorry. So you see that it has a very slim stem with purple flowers, and this is what it happens after a single cycle of herbicides. This plant mutates, puts dozens of stems together, and becomes resistant to herbicides for generations. And this is like a frightening reminder of how a resistance to antibiotic arise in clinical practice. And well, the floral ecosystems are very nice to think about ecology, but actually in the gut, everything is dark, is humid, and there's a very strong competition among microorganisms for nutrients. And it's really hard and difficult to study how these microbes interact between them, and this is probably one thing we need to study more in the field, and some groups have already started to study the prey-predator relationships among bacteria. And last is even more important to understand what these microorganisms are actually doing, because this is what actually matters for clinical practice. I mean, are these bacteria leading to positive or harmful effects? And actually, there's a very strong interplay between the microbiota and our body, and the immune system is one of the strongest interactions. I won't dissect this in detail, but the key message is that there are several components of the gut microbiota that are able to induce the Th17 response. This is like an accelerator in the immunity. It promotes the response against microbial invaders. And actually, it's been seen that the microbiota is able to amplify this response and to amplify autoimmunity. But in contrast, other components of the microbiota, they do just the opposite thing. They are potent T-regulatory inductors, which is like an immunological brake. And among them, there are salient bacteria, such as the short-chain fatty acid producers, which digest the dietary fiber into these metabolites, which are able to induce the Treg response, so decrease autoimmunity and the Th17 response. Actually, these interactions are very robust, and it seems that they have very strong implications, and probably behind these immune interactions, there's the link between the microbiota and many diseases, including autoimmune diseases, cancer, metabolic diseases, atherosclerosis, and it's also true that the microbiota is sometimes oversold, but there are people who have even suggested that the bacteria can even influence how do we feel like and who do we like. So that's something to take in mind. So what are the changes throughout life in the microbiota? So we're born with a very highly diverse, we are born free of bacteria, but we are soon colonized by a multitude of bacteria, and there's a strong immunological and nutritional pressure to select some bacteria. And over life, our microbiome changes following physiological changes, following perturbations, antibiotic treatment, diseases, sedentarism, diet, and so forth. And at the end, the microbiota of an old individual shows a very high variability among individuals. The alpha and beta diversity is different compared to younger individuals, and in some there's decreased firmicutes, and among them decreased short-chain fatty acid production and increased bacilli, although the functional consequences have not been well addressed as far as I know. But it looks like there are different microbiomes in the elderly, and these microbiomes correlate with frailty, which is interesting from a clinical perspective. There's even a microbiota signature associated with frailty, predominance of Egeritella and depletion of Faecalibacterium, which is one of the main short-chain fatty acids producers. These bacteria have also been linked with inflammation and also with neurocognitive dysfunction, even in studies in twins with and without dementia, which is really impressive to do as an study. And what's the link with HIV? well, HIV, we all know well the story. The epicentrum of the inflammation is probably the gut. The gut is damaged very early after the infection, and this results into loss of these Th17 cells and loss of the enterocyte barrier function. And probably because of bacterial translocation, as Julia explained, we've got this activation of the innate immune response, which has been linked with comorbidities. So there are different components of the microbiota which have been shown to influence these pathways, but also it is reasonable to assume that if we have such a strong perturbation in the habitat of these bacteria, we will see changes in the composition and in the function of these bacteria, which may promote this vicious circle of inflammation and viral replication. So in the past years, there have been a lot of studies trying to elucidate which is the microbiome of HIV-infected individuals, and there are actually great methodological issues about controlling confounders. So the summary is that there are some bacteria that seems to be enriched consistently across studies in HIV-infected individuals. HIV-infected subjects have increased abundance of pathogenic bacteria, pathobionts, increased proteobacteria, and these are changes that we see in the elderly. And there are a lot of studies where we saw increased Prevotella, and this is like the rainbow bacteria. This is a great contribution from Ursicaisa, because they found that this bacteria is enriched in MSM and seems to be linked to sexual preferences. So it was a main confounder in our studies. And in contrast, there is depletion of lactobacillus and bacteroides, which is short-chain fatty acid producers, which is something we typically see also in the elderly. So there are some similarities between the HIV-infected dysbiosis and the gut microbiota changes in the elderly. So I'd like just to give you an overview of how and why this could be relevant for HIV care. So we all try to think about the gut when we hear about the microbiota, but there are actually a lot of or some papers analyzing the microbiota in other compartments in which they've been shown that there's a very there's influence of the microbiome in inflammation at this site. So something which is very intriguing is the blood microbiota. This hasn't been studied well because of methodological issues and contamination with PCR, but there's a paper in HIV-infected macaques showing that early after HIV infection, there's a shift in the bacterial products that they are translocated to the gut, and surprisingly, there's a peak of bacterial translocation that precedes 1 week the peak of viremia, suggesting that the bacterial translocation amplify inflammation and the HIV target cell recruitment. And actually, they were able to find some HIV controller macaques and to induce the loss of virological control by inducing bacterial translocation. So it is likely that bacterial translocation is promoting viral replication since very early stages of the disease. And what we've seen, this is unpublished, is that there is also a microbiota signature in the blood of HIV-infected individuals with predominance of proteobacteria, and there are biomarkers of immune recovery. When we look at these bacteria in naive patients and we follow them for a year, we see that enrichment for lactobacillus predict better immune recovery, and depletion of actinobacteria is also is a risk factor for a poorer immune recovery. And these bacteria correlate with inflammatory markers, so this is something that probably deserves to be studied further. The other compartment is the vagina. This is really interesting because We already knew that there's increased risk of HIV acquisition, not explained by sexual risk behavior in many parts in Africa. And Linda Mason group found that this seems to be predicted by increased cervicovaginal inflammation. Many pro-inflammatory pathways were induced in the cervicovaginal fluid of the women who became infected later on. And Melissa Nattar was able to explain the cause of this inflammation. They found that the microbiota in women is mainly distributed in 4 cervicotypes, one with predominance of Lactobacillus crispatus, another one with Lactobacillus inner, and 2 of them with predominance of anaerobes, Gardnerella and Prevotella vivia. And basically, they found that when the main component of the vaginal microbiota is Prevotella, there's induction of a lot of pro-inflammatory pathways, recruiting presumably HIV target cells. So this is very exciting because we are facing a modifiable risk factor for HIV acquisition. And the penis is a neglected site in the microbiome research, but there's been papers also looking at what happens after circumcision. We already knew that circumcision protects of HIV acquisition by men. So they were able to demonstrate that after circumcision, the anaerobes drop in the penis, and the proportion of anaerobes correlate with the risk of HIV acquisition with the concentrations of IL-8, which is a very interesting cytokine because any cell with toll-like receptors can produce this cytokine, and it mediates the recruitment of neutrophils. So again, we have bacteria anaerobes that are promoting inflammation and the recruitment of HIV target cells, and this is also a modifiable risk factor. So the lung is being less studied, but there are very interesting papers showing that the risk of the increased risk we see of bacterial infections and tuberculosis in HIV-infected patients may be explained by induction of regulatory T responses suppression, the specific immunity against tuberculosis, and this seems to be mediated by the production of butyrate by the anaerobes. It seems also that there are alterations in surfactant production mediated by impaired metabolism of the precursors, and there's also decreased cellular immunity linked to bacterial pneumonia, which has been explained by altered linolate metabolism. So in sum, we have some pathways which could explain something that is not explained today, which is why suppressed patients with good CD4 counts have increased risk of tuberculosis and bacterial pneumonia. And this could even, although this is speculative, why we see so dramatic COPD disease and increased risk of lung cancer not explained by smoking. So the butyrate leads us to the gut, which is the better studied compartment. So I just wanted to outline 3 pathways by which the microbiota can influence inflammation in HIV. One, as I mentioned before, is the short-chain fatty acid production. So these metabolites are produced in the gut by some bacteria, and among them, Faecalibacterium prausnitzii is a major butyrate producer. This is a very dominant commensal in the healthy gut, and it's able to digest the dietary fiber into this small molecule, the butyrate, which is very interesting because butyrate is the main source of energy for the colonocyte, is an anti-inflammatory molecule, and is also even able to inhibit the histone deacetylases. Martha talked about them before. And this is kind of a remote control of gut bacteria of our gene transcription activity. And we actually found that HIV-infected patients have decreased abundance of these bacteria, decreased levels of butyrate, at least in our study, and this correlates with the extent of histone acetylation in blood, suggesting that this is one mechanism at which the bacteria could be influencing inflammation and immune recovery during ART. The second pathway is the production of trimethylamine. This is a metabolite, also, which is also derived from the diet, from phosphatidylcholine and carnitine. Some bacteria produce this metabolite, and in the liver is oxidized and is a very potent protherogenic metabolite. And vegans actually don't produce this metabolite. So there was the hypothesis that maybe this dysbiosis in HIV could influence the increased risk of cardiovascular disease we see in our patients, and at least one study had pointed before that the plaque burden in HIV-infected patients is well correlated with the concentrations of these metabolites. And a few weeks ago, a paper from a German group focused on the microbiota of HIV-infected patients with and without cardiovascular disease, and they found up to 50 bacteria who were significantly different, more abundant or less abundant in the population of patients with cardiovascular disease. And the third important pathway is the the tryptophan catabolism, and this was one of the first papers published in this field in 2013. So this enzyme, the indolamine deoxygenase, is constitutively expressed in macrophages and dendritic cells in the mucosa, and it's able to catabolize the tryptophan into other catabolites which are immunologically active. They induce the Treg response, so they allow for microbial translocation, and actually the biomarker of this pathway, the kynurenine-tryptophan ratio, is a strong and independent predictor of mortality in HIV. So the San Francisco group was able to identify in HIV-infected patients up to 140 bacteria encoding the enzymatic machinery to reproduce this human pathway, and actually this correlated with inflammatory markers. And in a different study in elite controllers, it's been shown that this pathway is preserved in elite controllers. So it is probably very relevant for HIV immunopathogenesis, the effect that the microbiota can exert on this pathway. So the bad news is that most of studies have focused on the sequencing, 16S sequencing data, and this allows you to understand who is there, if you have Ewoks or imperial soldiers. But to know if these imperial soldiers are doing something bad, you actually need to go deeper into the functional studies, and you may want to study the transcriptome, the proteome, or the metabolome, so that you can deduce if the gremlin is quiet, or you have fed him after midnight and it has become into a bad creature. So for this you need other kind of studies, and this was our first study in the field. We just looked at the metagenomic data using metagenomic shotgun to understand what genes were more represented in the HIV-infected populations. And I think the first thing I learned in this field is that dysbiosis is not necessarily a bad consequence in HIV, because we found that, okay, there was high abundance of some pathways belonging to inflammation, LPS synthesis, but there was also enrichment in the HIV-infected patients for antioxidant pathways, synthesis of cofactors, vitamins, suggesting that the microbiota is trying to repair a harm is going on. And this has been a very nice contribution from Irsi Kaisa group in a very beautiful paper in which they have also profiled all the genes in a very fair amount of patients. And I think their interpretation is something similar. They found that patients with very low nadir CD4 also have lower gene counts in their whole microbial communities. And this correlates also with an adaptation to oxidative stress and to inflammation. So we must be cautious about interpreting this biosis as a bad consequence of a disease, because actually it could be also an adaptation doing beneficial effects on the host. And this is a different approach. This is a proteomic study. When we compare different diseases and healthy controls, we found that in the fecal microbiota, 80% of bacteria in healthy subjects are dormant or died. And in contrast, in C. diff infection and in HIV, a large proportion of bacteria are activated. And when we did metabolomics, we found that some bacteria seems to be contributing to immune repair, to immune recovery by acting as anti-inflammatory buffers, accumulating within the bacteria pro-inflammatory mediators or anti-inflammatory mediators such as biliverdin or oleamide, while in subjects who are not responding well to ART, these bacteria are not acting as anti-inflammatory buffers. And I think this is very interesting since it gives you some insight on how the bacteria could be influencing immune restoration under ART. And this is hard to understand, even for those who are involved in the papers. But another approach is to try to integrate different omic techniques, and here we try to combine 16S data with transcriptomics and metabolomics to see which pathways depend on what bacteria. The findings, I think, are consistent with the previous message. We found that there are some bacteria which seem to be contributing to inflammation or expressing pathways related to resistance to oxidative stress, but there are also other bacteria which are doing just the opposite things. They are they are resistant to inflammation, they are anti-inflammatory, and they correlate with markers of T-cell activation. So a different approach is you take this bacteria and you go to the lab and see what happens when you do immunostimulation experiments. And this group from Colorado has been able to purify the bacteria from the stools of HIV-infected individuals, and they've seen that when you compare what happens in monocytes when you stimulate them with bacteria from HIV-infected individuals as compared with bacteria from HIV-uninfected, the activation of monocytes and T cells is much higher when with the microbiota from HIV-infected individuals, suggesting that this microbiota is actually pro-inflammatory, and they've also been able to see it doing fecal transplant with the microbiota from HIV-infected and uninfected MSM to mice, and they've seen consistent data showing that fecal transplant from HIV-infected individuals elicit higher T-cell activation than when we use the stools from HIV-infected individuals. So I believe that clinical trials are necessary to fully understand what's the feasibility of manipulating the microbiota and the clinical consequences. We probably have different endpoints related to lung diseases, to genital tract problems, cardiovascular disease, to GI tract, and to inflammation and HIV immunopathogenesis. And we may want to use prebiotics, probiotics, phages, antibiotics, fecal transplants, or to selectively use the bacterial metabolites that seem to be playing a role. So we've tried prebiotics, and it was surprising to see that when we looked at the effects of prebiotics 6 weeks in the same patients by 16S sequencing or by proteomics, we see a different picture. Using 16S sequencing, we detected some changes only in ART-naive individuals, but using proteomics, we found that in all groups, HIV-infected, uninfected, treated, and naive, there was a very strong activation of bifidobacteria, although we were unable to see changes in inflammatory biomarkers after this 6-week intervention. So we decided to do a larger trial, combining in a high-dose cocktail of prebiotics, probiotics, oligonutrients, omega-3 and 6 fatty acids and essential amino acids, which were given for 1 year to late presenters, since we believe that this was a population that may benefit from from boosting the immune recovery. And it was great to see that ART is really effective, all the biomarkers improved, but it was a bit disappointing to see that we didn't achieve anything in terms of the probiotic cocktail compared to the placebo. There was no signal of an effect of markers of inflammation, bacterial translocation, T-cell activation, and we didn't see even changes or only very small changes at the gut microbiota using 16S sequencing. So we were a bit frustrated, but there's probably another way to try to deal with microbiota manipulation. And Samsuk Ma and Ivan Vukovic in San Francisco did a small trial with 6 patients using fecal transplant via colonoscopy. And it was also a bit disappointing to see that the extent of engraftment is very limited compared to what you see when you give FMT to patients with C. diff diarrhea. This means that you achieve fecal engraftment from the donor, and this means that the extent of engraftment was very limited. So we are facing probably different problems. C. diff infection is like an arid field. There's no competitors. There are only some weeds growing up. So it's probably easier to repopulate this field than if we go to HIV or to chronic diseases. This is the Monegros Desert, very close to here. And here, what you have is an ecosystem which is adapted to a arid and continental and extreme climate. And you can't pretend to plant here a tropical forest because simply the conditions are not the conditions you need. So we are going to be we are going to need to be very creative for interventions. So we designed this trial. This is fecal transplants by oral capsules from 3 selected donors based on their microbiota, and we are trying to reseed the field little by little to introduce changes sequentially, which is something that ecologists always say that it's very important to change any ecosystem. And we are just now analyzing these results. The trial has been completed. So as take-home messages, we probably need to think more about the microbiota as an ecosystem. It is clear that it's a very strong immune modulator. There are similarities between the microbiota and HIV and the elderly. It's been linked with T-cell recovery and inflammation during ART. It influences by promoting inflammation, HIV transmission in men and women. It affects the risk of lung diseases. But we need to understand better the functional implications. We need to assess more interventions, and it's either way a really exciting area of research. I'd just like to thank all my collaborators in different parts who are contributing to these studies, and thank you for your attention.
Okay, thank you very much for that interesting talks. I learned a lot, and if you have any question?
Question for Sergio or Victor. I wonder if there are trials looking at the effect of providing short-chain fatty acids.
Ventura, could you use the microphone? Thank you.
Let's rewind. Great talk, Sergio. I wonder if you have assessed the acids, high dosage might be improving the immune recovery. And then probably what happens is that we are always thinking of this at the very end, instead of starting this intervention very early, even in those which haven't had significant impact in the immune CD4 count, but in that setting even we could even improve much better or faster speed up the recovery of CD4s. And I want you to speculate about that.
I think they are both great questions. Indeed, we may be trying to change the microbiota, and we may simply want to use the correct metabolites, driving the beneficial effects. And I think there's a debate. The short-chain fatty acid is not clear to me totally in HIV. Maybe Alan may want to add more information because he has studied it. But it will be a very nice proof that these metabolites are actually good or not, or they are simply intermediate factors in a cascade of other things we are not seeing. So I've thought a lot of times in doing a trial with butyrate, but I haven't had the time. But it will be very interesting. And regarding the other question, it's totally true that the window to change the microbiota maybe, the best moment is early in infancy because everything looks like it's very hard to change an ecosystem when it's well established. And actually when in the exploratory study we did with prebiotics, one of the main conclusions was that in ART-naive patients, there's some possibility to introduce changes, but in ART-treated patients, it was really hard. So we didn't see actually an effect. I mean, just when you are looking at everything, you pick up things, but there was no clear effect in ART-treated patients. So we may want to assess longer interventions, and the problem is that it's not a problem, it's good, but the ART-naive patients are today very difficult to include in trials because we put all of them on ART very soon, so it's going to be something difficult to prove.
Just one question. We don't have time for more questions. The last one, please, and we can continue the discussion during the coffee.
Great. Well, again, a great group of presentations. I'll comment on the question of short-chain fatty acids, that the Australians have been studying butyrate. CSIRO actually makes butyrate capsules, and we've talked with them about trying to get those implemented for clinical trials. They are doing this actually in allergic children, and they are using them clinically in that phase of development. There's other data from Charles Mackay, who's done some really elegant work in using acetate. And I think we should not neglect the others. Butyrate is of interest, but there's also acetate and propionate, which also may be relevant as well. So I think those are good areas to pursue. I wonder for the whole panel about integrating, perhaps looking at the systemic immune system and how looking at senescence in the immune system or using immune cells, is that perhaps a sentinel event? Because sometimes it's harder to monitor in tissue or other sites. So I'm wondering, Marta and then Julia and Sergio, how you might think about using the immune system for monitoring that. And since we're at an aging meeting, I also asked Marta if she'd like to comment about the role of senolytics, which is getting a larger interest in the aging field.
Well, I'm an expert on senescence, on DNA repair, and on the DDR, but not on the use of these drugs. But I think it's very exciting. What it's getting more and more relevant every time is that senescence and the accumulation of DNA damage on cells, it's damaging a lot of mechanisms which are very closely connected. And for example, with the immune system, I see this is a complete correlation. And this must be the direction of research in many areas. But I don't know anything about the senolytics themselves because I don't work with these drugs. It's just that I thought it would be interesting for the audience here because you work with HIV. So if you have a more concrete question on senescence, maybe I can help you more. If not, I'm sure you can.
Yeah, this is a very, very interesting question. The problem is whether the immune senescent cells are exactly senescent or not. I'm not sure about that. We are measuring immunosenescence in periphery. We don't know if this immunosenescence is strictly the same at the lymphoid tissues, which is probably different. And the immune-senescent cells, they still may proliferate. We can make them proliferate.
This is what I thought, yeah. Why? How?
These are real senescent cells. We call them replicative senescent cells to the CD28 negative, CD55-7 positive cells. But I think they can proliferate, so we can revert this senescence. So I don't know if they are truly senescent. I don't know whether they will be sensitive to senolytic drugs or whether they contribute the most to the general inflammation, or are other cells in the tissues that are contributing much more to this phenotype.
In this regard, if a senescent cell resumes to divide, maybe has inactivated some of the control pathways like P53 or P16, and then it's able to gain again the replicative, and this is also very dangerous, but this can be checked, this is easily checked. If not, maybe it's just an old cell, pre-senescent cell. I don't know.
I'm sorry, but we don't have more time. We can continue discussing during the coffee. Just 10 minutes because we are late, and 10 minutes, just a coffee and come back. Thank you very much.
Eureka. Good afternoon. My name is Cécile Tremblay. I'm professor of infectious disease and microbiology at Université de Montréal, and I will be presenting the next 3 speakers this afternoon. But before I do that, I was asked to tell you that after this session, there's going to be a photo group. So we're asking all of you to gather near the main entrance where the photo will be taken before you go to the next break. Is that okay with you? So the first speaker is Dolores Villas. She's in the Neurodegenerative Disease Unit here at Hospitals Germans Trias y Pujol, and she will be discussing neurodegeneration. The second speaker is José Ramón Blanco from Hospital San Pedro, Center for Biomedical Research of La Rioja, and he will be discussing muscle decline and sarcopenia. And our third speaker, Valeria Fink, in clinical research of the Fundación Huésped in Argentina. So it's going to be more clinical science this session, so enjoy. And then we will have the discussion at the end of the 3 talks. So, Dolores?
Well, good afternoon, everyone. Thank you for inviting me to be here with you today. Well, I'm a clinician, I'm a neurologist, so we will totally change the point of view from the basic science to the clinical point of view. And I will try to review the age-related neurodegeneration, which is a complex topic, but we will try to focus mainly, in 3 aspects. First, we will briefly review what neurodegeneration is, and then we will focus on normal human aging human brain. Basically, we will review some important aspects of the neuropathology of the normal aging human brain, and then we will also review some important aspects of the clinical expression of the cognitive changes in the elderly. In the last slides, we will remark some important points of the neurodegenerative diseases. Well, we all get older. Even this beautiful girl probably you remember her, was the cover of the National Geographic journal some years ago. And our brain also gets older. How? Well, all you know, the previous talks, my colleagues before say some things about what happens in cells during the old age, but neurodegeneration is the progressive loss of function of neurons, and including the death of the neurons. And all the cells in the central nervous systems are affected by age. But there is a huge variability between people, between cells, and between how people compensate the deficits of the senescence in the brain. Well, as my colleagues say before, I don't know. The cells in the brain are damaged by oxidative stress, DNA damage, inflammation, etc. And this is the scenario that this is the scenario we have in the old age. But in this scenario, we can have some risk factors or some protective factors for neurodegenerative diseases. For example, we can have some genetic risk factors for Alzheimer's, like ApoE4. Also, we can have some genetic mutations associated with neurodegenerative diseases. For example, we can have a mutation in alpha-synuclein gene, which is a risk factor for Parkinson's disease. We can have also environmental risk factors for these diseases, such as, for example, physical and mental inactivity that we know that is a risk factor for Alzheimer's. But we can be in the other side of the slide. We can have some neuroprotective factors that in this scenario, even if we have all these, we will never develop a neurodegenerative disease. The reality is that when we get older, all we have a loss of our brain volume, especially after the age of 55, 60 years, our brain becomes atrophy. In fact, in the last year, we have several studies with functional and structural MRI, and we can see that different areas in the brain have different risk of being more atrophic. For example, we know today that the prefrontal cortex, the more frontal part of the brain in the frontal lobe, is one of the most affected areas of the brain by age. Also, we know that the white matter, especially in the frontal lobe, is very, very affected with age. And we can also observe some regional blood flow and some other abnormalities, for example, in the functional connectivity in the brain with age. If we can see a brain, a microscopic brain, we know that when we are old, this brain has a lot of atrophy. And we can also see this in an MRI. And this slide, I think, is very important because we can have 4 different scenarios when we are old. Here we can have a normal, a healthy aging without brain pathology, with normal cognition. In the other side, we can have abundant pathology, neuropathology, but abnormal, and also abnormal cognition. This is a cognitive decline or dementia. But I would like to focus your attention in these 2 parts of the slide. We can have this situation. We can have a normal cognition with age. We can have an old person with normal cognition, but with abundant pathology in their brain. Now we will focus in this situation. And we can have in the other part of the slide, we can have an abnormal cognition, but without pathology in the brain. That is what we name cognitively frail aging. We know that with age we have some neuropathological changes very commonly. First, cerebrovascular disease. It's very frequently, especially in very old ages, we know that it's very frequent to have clinically silent cerebrovascular events. For example, this very small infarct that we call vacuum infarct. These are very common. We can see these lesions in an MRI. You can see that even almost 80% of people over 80 years old can have white matter hyperintensities. We can here observe some of these in the MRI. These are very common. We can have these lesions even with 20, 30 years old, but they are even more common when we get older. And if we could get a microscope examination of all old people, we can see that even almost 90% of people over the age of 90 have these brain lesions. So in some way, we can consider that cerebrovascular lesions are very, very frequently in old age. Almost could be a normal condition during the elderly. For example, here we can observe some studies where we can see that since the 5th decade of life, it's very frequent to have this, and especially in the 8th and 9th decades of life, it's very, very common. Another important situation that it's more recently, we know now that old people have a very increased frequency of hippocampal sclerosis. For us, for neurologists, hippocampal sclerosis is synonymous of epilepsy. It's very frequent in young people, epilepsy secondary to this situation, hippocampal sclerosis. But now we know that in old people, especially in extreme old people, this hippocampal sclerosis is also very common and is related with cognitive decline. The hippocampus, as you know, memory is in the hippocampus here. And in the case of old people, we see that here in the hippocampus there is deposits of a protein, which we call TDP43, which is a protein that we observe in frontotemporal dementia and other neurodegenerative diseases. So now we know that also in a normal elderly, we can also observe this hippocampal sclerosis, and that is strongly correlated with cognitive decline. But we also, today we know that it's very frequent also in the elderly to have protein aggregation. As we will review in the next minutes, the neurodegenerative diseases, now we try to classify them as proteinopathies. We know that Alzheimer's is a amyloidopathy with deposits also of tau protein. We know that Parkinson's is a synucleinopathy with deposits of alpha-synuclein. But in the last years, there is some studies, some neuropathological studies that show us that we can also see these abnormal proteins in the brain of elder people. For example, it's very frequent to have beta amyloid, which is one of the main proteins we observe in Alzheimer's, but we can also observe beta amyloid in almost 70% of elder people without cognitive impairment. Other proteins like alpha-synuclein, which is the protein Parkinson's disease have in the brain, and also tau, almost 25% of cognitively intact elder people have these proteins in the brain. Even more, it's very frequent in old age to have copathology, to have mixed pathology, some mixture of these proteins in the brain, even with a normal cognitive performance. So it's normal to have a cognitive decline when we get old, so the answer is no. We can have, it's not common, it's not normal to have a cognitive decline when we get old, but we can have impaired some basic cognitive functions like attention, memory, and language processing and decision making. We can have some deficits in this basic cognitive function, but not in other ones. For example, it's common. I don't like to bore you with this, but for example, it's very common when you are old to have some problems with selective attention. For example, it's common to have some problems if you try to focus in one thing, trying to avoid all the other things. For old people is more difficult than for young people. We can have with age also problems when we have to attend to 2 or 3 or more different things. For old people is also more difficult than for young people. And we can have also some problems with working memory. Working memory is, for example, the part of the memory we use for remember, for example, a telephone number, or to remind, or to try to remember a number backwards. This is working memory, and this is normal to have some problems when you get old. But not all other kinds of memory or other parts of the brain. So we can have with age some slight problems in some basic cognitive functions, but it's not normal to have a cognitive decline with age. And it's important to know the concept of the cognitive reserve, because all people with in old age have all these problems in the cells we previously see, like mitochondrial damage, like DNA repair problems, but not all people have cognitive decline. So one hypothesis is the concept that the cognitive reserve is the ability of each individual to try to solve or to compensate the problems that we have in the cells of the central nervous systems with age. For example, and this is, there are some factors that make that our cognitive reserve is higher. For example, high level of education or participate in cognitive activities that increase our cognitive reserve. And this is very important because, for example, here we have two patients. Patient 1 has a higher cognitive reserve than patient 2, and both have the same brain damage, for example, lung stroke. And immediately, this patient 2 with less cognitive reserve goes down the threshold for a cognitive impairment. However, this patient can still be cognitively intact. Also, for example, if we have these two patients and both have Alzheimer's, the patient with a high cognitive reserve needs to have more neuropathology in his brain to get the cognitive impairment. It's for that, and this is in the last years, it's a trending topic, it's very important, and it's for that that it's very important to diagnose the cognitive decline in general in people and to do neurocognitive rehabilitation. Tomorrow, the neuropsychology, we will review this topic, and this is very important to try to give our patients the best management we can give them. So, dementia is not a normal part of aging, but it's a huge health problem today and affects not only the patient but also the relatives, and it's a very, very huge economic and political problem today. For example, it's estimated that for 2050, the number of people with dementia will be triplicate in the world. And especially in very old people, the most common dementias are Alzheimer's, which is the most common one, and also the vascular, mixed dementia and frontotemporal dementia, but Alzheimer's is, of course, the most frequent one. In fact, the World Health Organization and some important commissions, like the Lancet Commission, has dementia as a priority in the last year. But we can have also different neurodegenerative diseases where we can observe different proteins in the brain, and we don't know why is that. We don't know why in Alzheimer's the cells most affected are those from the hippocampus. We don't know why in Parkinson's the cells more affected are the dopaminergic cells in the substantia nigra. We don't know why in the amyotrophic lateral sclerosis we have another different cells most affected. So we don't know why, and we don't know why in Alzheimer's we have tau and beta amyloid, and we in Parkinson's have alpha-synuclein aggregates in the brain. We in neurodegenerative diseases still have a lot of questions without an answer. For example, probably you know sometimes this is a Levy body, which is an abnormal aggregate of alpha-synuclein in a dopaminergic neuron. Here we can have the typical deposits of beta amyloid in the brain of Alzheimer's people, and the deposits intraneuronal of tau also in Alzheimer's, etc. So now this is the way we see to neurodegenerative diseases. Now we, Still now, but especially in the last decades, we normally see the patient, we diagnose the patient with a syndrome. For example, well, a patient has a cognitive decline with primarily affected the amnesic function or with the allogopenic variant. So I think he or she has Alzheimer's. Well, this patient have, for example, tremor, rigidity, this patient have Parkinson's, but there is a huge overlap between neurodegenerative diseases, and sometimes we don't know what the patient have. Sometimes we can misdiagnose a patient with Alzheimer's and with Lewy body dementia, for example, and this is very common. This happens every day at the clinical practice. So in the last year, as we know each more time more about the neuropathology of these situations, we are going to this part of the slide, and we are going to try to diagnose if we are in amyloidopathy, like Alzheimer's, or if we are talking about a synucleinopathy. But the question is that today we don't have a biomarker to try to detect these proteins in the people, in the patient. In the last year, probably you heard about PET of amyloid and Alzheimer's and biomarkers in the cerebrospinal fluid in Alzheimer's, and this is very, very useful, and we are now in the routine clinical practice using these biomarkers, but we are still in the first stages of the research in other kinds of neurodegenerative diseases. For example, it's being developed the PET for tau or PET for synuclein, but we are still in the first stages of the research in this sense. And it's not the topic of my talk, but in HIV patients, it's very common to have a cognitive decline. And if you can see, the problems are similar to those that we observe in all people. For example, it's frequent to have problems in attention, problems in working memory, and also other problems. And why is that? Well, you know better than me. It's because of the direct effect of the virus in the nervous system. It's very frequent also in HIV patients to have cerebrovascular disease, and there are also other indirect factors like the use of drugs, and also the fact that the drugs we give the patient, the antiretroviral drugs, also can help to the cognitive decline. But now we have to take into account that HIV patients are also old, and they can also have Alzheimer's, Parkinson's, frontotemporal dementia. This is a new scenario. Before, probably all the patients with HIV and cognitive decline have this condition, HIV-associated neurocognitive disorders, but probably now we will start seeing patients with HIV and Alzheimer's and Parkinson's. So I think it's very, very important to assess the patient with a neuropsychological test to diagnose which is exactly the problem with the cognitive decline of the patient, to try to diagnose better the patients. And it's very important to start the neurocognitive stimulation early, and the treatment the patient needs for the specific disease we can diagnose. So my take-home messages are that neurodegeneration affects everyone, but there's a huge variability among individuals, that aging human brain is not synonymous of cognitive decline, that we have some conditions like cerebrovascular disease or hippocampal sclerosis, but we can have also, that are very frequent in the elderly, but we can have also abnormal protein aggregation that we observe also in neurodegenerative diseases. And finally, that is not normal to have a cognitive decline with age, and dementia and other neurodegenerative diseases are not part of normal aging, and it's very important to assess the patient properly to try to diagnose them better and to give them the management and the treatment better than we can. Thank you for your attention.
Well, good afternoon, everybody. First off, I want to thank Eugenia and the rest of the members of the Organization Committee for her kind invitation to stay here with all of you. Well, why is important muscle? Well, after preparing this session is very important for multiple reasons. First, about muscle mass is about more or less 50% of all the human body mass, so it's really important. From a functional point of view, it's very important in motility and stability, and also muscle plays an important role in some metabolic factors such as glucose metabolism. Muscle structure is very complex, but the most important for us could be to define the presence of different types of contractile proteins that are implicated in mobility, stability, and other activities. For example, the first one, type 1, are very rich in mitochondria, so it produces a lot of energy, and for this reason, and for the structure, is very important and very resistant to fatigue. This type of protein is able for running, for example, marathon. On the opposite, we have the type 2 proteins that are with low amount of mitochondria, and for this reason, the resistance to fatigue is very low, and is only for running short period of time, for example, on sprint. How to define sarcopenia? Sarcopenia is not easy to define. No? Some authors consider that it's a syndrome with a progressive and generalized loss of muscle, but they consider that's necessary at least one more criteria. This criteria could be the low muscle strength or, well, a low physical performance. It's very important to take into account that even in the case of practicing exercise every day, every year, at the end, our muscle will suffer atrophy. So this is a natural process related to aging. Some more consideration. Well, sarcopenia would be to the muscle like osteoporosis to the bone. I think that this idea is very important for summary the importance of sarcopenia. Some authors consider that muscle is very complex and very important, and for this reason they say that probably it should be considered like an organ for their importance. And as always, muscle quantity and quality is not the same. This is very important also speaking about muscle. Weight is also important. This is a real public health problem for several reasons, and all of them are a negative impact. For example, this patient will have a poor outcome, a higher risk of physical disability, more osteoporosis. Osteoporosis and sarcopenia are very close to, and for this reason sometimes it's similar talking about this topic. High risk of falls and fractures, and this patient will have a poor prognosis that other patients not having sarcopenia. How to define sarcopenia? Here we have a very huge problem because there is no consensus about how to define sarcopenia. These are proposals of different societies. As you can see, these societies are employing different criteria and different methods for defining sarcopenia. Some of them are employing computed tomography, other one DEXA, gait speed 1 meter second, other one 0.8 meter. So this is a real problem because we are not having a complete definition. The real impact of this disease is not able to be well understood. Let's see what happens if we employ one of the definitions. For example, employing these criteria in different countries, we are able to identify that in some cases in Japan, for example, is moving from 2.3% to USA 36%. Probably it could be related to lifestyle, age, a lot of conditions. However, what happens if we try to analyze all these criteria of the different societies into the same court? We are observing that we are moving from 5.3% to 1.3%. This is a very high differences in prevalence, so this is a problem. However, even in the beta situation, we want to translate this to a worldwide problem, that sarcopenia, about 5 million people are affected by sarcopenia. So this is a real important health problem. In case of getting a better consensus definition, it could let us to understand better sarcopenia. However, in spite of this situation, multiple factors has been implicated in sarcopenia. For example, some of them are genetic, hormonal, metabolic, the age, and this conduces to a reduction in muscle mass and strength that leads to sarcopenia, that aggravates weakness, that again produces reduction of physical activity, that again sends out to the rest. So we are in front of the vicious circle that increases and produces continuously sarcopenia and is very negative for our patients. Let's see some of these questions. No? It's known that men have more muscle mass and strength than women. However, after the 30s and mainly after the 70s, muscle mass and strength decline. So this is a natural process. It's not a process related to another diseases. It's only a natural process. As you remember, before I commented about the different types of contractile fibers in the muscle. Well, as you can see, a young muscle, there is a balance more or less between the fibers type 1 and type 2. During the aging, we are losing type 2 fibers. And in sarcopenia, practically type 2 fibers don't exist. The reasons are not well known, but this is very important because type 2 fibers are mainly present in ears and lungs. So this could be a reason for sarcopenia and the implication there is this. The reason could be multiple. For example, aging, but it's also important the relationship with inflammation and hormonal situation and condition that could increase the loss of type 2 fibers. What about diet? Well, diet is very complex, contradictory articles, but in some cases, most majority of the authors have observed that the concentration of protein is related to the muscle mass and strength. As you can see, having more protein, you are able to get muscle mass and mass strength. Something similar is observed with some vitamin D, vitamin C, and with the intake of some other products like omega-3. So diet plays an important role here too. What about lifestyle? Well, I show you here two meta-analyses. One of them is focused on cigarette smoking, and the other one on alcohol consumption. The cigarette smoking, the authors observed that it provides an association with sarcopenia, but it was not observed in relation to alcohol. Probably there is multiple bias that can influence in this result, but this kind of study are necessary in our patients. Obesity is well known that it accelerates aging, so it's an important factor too. Again, not only but we are moving lower or less, it limits our physical activity, but also is increasing some cells, macrophages, senescent cells, more inflammation, again sarcopenia, less physical activity, and again we are in presence of another vicious circle. So this is very common in this process. Well, sarcopenia is not only the loss or the reduction of the muscle mass and strength. It's important the role of the fatty tissue. It's not only important what happens around the muscle, but also inside the muscle. This is a term that is now defined as myosteatosis. Myosteatosis is important not only because it's implicated in local and general inflammation, but also because it's implicated also in fiber muscular. The most important action is the activation of proteolytic system that produces changes in metabolic and mechanical places that around and produce and increase the risk of the cardiovascular risk, metabolic problems, and also a functional decline. Well, if there is important the role of the proteolytica in this balance, although we have to find anabolic situation. Anabolic situation will try to increase the muscle mass. What happened? Probably is happening that during the aging we are losing our anabolic situation. So this could be an imbalance in favor of catabolic situation. So very important, the inflammation and myostatin. To know, to understand the role of the importance of inflammation, as you can see, red line is putting us in focus what is happening for our muscle wasting. On the other hand, in red line, what try to improve our muscle mass and reduce the muscle wasting. Very important exercise, but also, as we could see in the diet, the protein intake administration. Myostatin. Myostatin is very important and interesting protein. It's a protein that's produced by the muscle and this protein limits the growth of the muscle. Your left, this is a current mouse. It's your muscle. And after blocking myostatin, after blocking this protein, this is the new mouse. It's very hypertrophic. There is an important increase of muscle, just like Popeye's mouse. Well, what is happening in different studies trying to get into the clinical and to improve the situation of our patients? Well, in animal models, when you are giving to the mouse anti-inflammatory drugs, they improve their anabolic situation. In human, it's just the opposite, but the result is the same, because decrease the protein breakdown. The consequences is the same. Our patient improve. But what is happening in elderly? Elderly is necessary not only this anti-inflammatory drug, but also training, resistant training for improving. However, even practicing the same exercise, the same intensity, the same training, aging patient will recover less muscle mass for multiple reasons. It's because in younger, muscle protein synthesis and breakdown are in a balance. However, during the aging, for different reasons, inactivity, inflammation, other one, muscle protein breakdowns is over muscle protein synthesis. For this reason, I told you, I commented at the beginning of the session that some processes are related to a natural process of aging. 2 slides, very, very quickly, only for show you what is trying to study. The role of inflammation, very important. The results partially in some cases, in others no. And proteolytic pathways. In some cases, no results when we translate the animal models to human, and multiple studies need to be performed because so far there is no data in human. And this is the pathways mainly implicated in this process. Very important, mTOR, as you know, is related to rapamycin. Rapamycin is an important drug for improving aging and long survival. It's not my intention to review this treatment, but it's important to note that some of them has described the improvement of some patient in relation to sarcopenia. This drug also can explain in some way different ways or pathways for working on sarcopenia. Anyway, currently no agency has approved none of them for treating sarcopenia. So we have a problem here. Probably now are working with inhibitors of myostatin probably for improving this situation. We are talking a lot about aging, but it's necessary to remember that there are multiple conditions that could also produce sarcopenia. For example, cancer related to cancer, autoimmune diseases, and also frailty and sarcopenia are very close to and very important, both processes. Well, and what about HIV? Well, this our search looking for last week, Sarcopenia muscle, I found more or less 5,000 articles. When I included HIV, only 35 not being strictly, because we are strictly, it was less. This supposes more or less 0.6% of all the articles. So we have here a very interesting topic for research and for study. The main messages about HIV and sarcopenia. Sarcopenia, as well as other comorbidity or diseases, is more prevalent in HIV-infected patients. This is common to all the diseases that we are studying in HIV patients. Very important for their clinical implication. Those patients with less muscle mass will suffer from cardiovascular problem. This is an interesting article. And I enjoyed a lot reading this article of Patricia Echevarria Eugenia about sarcopenia and HIV. This is the factor that they found related to sarcopenia. A very interesting article last week, what published online was previously HIV-infected patient on stable antiretroviral therapy, undetectable viral load. All of them more than 350 CD4 cells. They observe compared to general population, the same age more or less, more inflammation and immune activation is known, so it's not a problem. The patient, HIV-infected patient, reported more self-report fatigue than seronegative patient. This patient also report the emerging of deficit in physical function, and the most important is this patient, when a biopsy was performed in the muscle, our HIV-infected patients are showing some results that are observed in aging seronegative patients. So something is happening also in our patient. And remember, the muscle is very important and play important role on metabolism. So this is a question that we have to take into account. In summary, sarcopenia is a public health problem that we have to improve because it's necessary for improving our patient situation. Even in the best of the cases, we are talking in general population that more than 50 million people are suffering from sarcopenia. Diagnosis is difficult, probably is necessary a biopsy so far, but probably if we are able to identify some biomarker, it could be very useful for studying a lot of population. More research are always necessary for every process. Prevention here is essential. It's necessary to explain our patient that they have to change lifestyle, and this includes practicing exercise. And in case of getting all the above, perhaps we could minimize this adverse impact. Thank you very much for your attention.
Well, good afternoon, everyone. This is the last presentation. I'm going to try to go fast because I know we are a bit behind. First of all, I want to thank the organizers, in particular Dr. Pedro Cana and Dr. Eugenia Negredo, for inviting me to this symposium. And now we are going to discuss age-associated cancers. So as we know, why are we discussing this? Actually, this is data from the United States. As you can see, most of the cancers are happening in this aging population. And this is data from the GloboCan from this year, and these are the most commonly found cancer among general population: breast, lung, colorectal, and prostate. That so far, it's not what is happening in the HIV population except for lung. So And what about cancer and HIV? We know that cancer and HIV had been very intimately linked from the very beginning of the HIV epidemic, if we consider that Kaposi sarcoma was a hallmark of the first AIDS cases in the early 80s. However, with the advent of combined antiretroviral therapy, people living with HIV started to live longer, and the profile of cancer in this population started to change. And nowadays, in those settings, when there's access to testing and access to care and access to antiretroviral therapy, cancer had become a very big issue for morbidity and mortality. It is estimated that around there's a 50% increased risk of cancer among people living with HIV. Data from some cohorts have shown that about 1/3 of this population will eventually die from cancer. So this is quite old, but it's quite clear in the first line. We have the 3 AIDS-defining cancers, Kaposi sarcoma here, non-Hodgkin lymphoma, and cervical cancer. And mainly for sarcoma, for Kaposi sarcoma and non-Hodgkin lymphoma, here when you see the big decline is when the combined antiretroviral therapy became available, and it's clear how these AIDS-defining malignancies started to decline. However, other malignancies, as anal cancer, lung cancer, or liver cancer, started to increase. And this is just to have an idea of how much more common are some of these cancers among HIV population. So we have in pink the first 3 with the AIDS-defining malignancy, mainly Kaposi sarcoma is around 200 times more frequent in this population, and it's nearly 80 times more frequent for non-Hodgkin lymphoma and cervical cancer. We are going to discuss later. Here it's 8, it varies from different series. However, some other cancers which are considered non-AIDS definings, like anal cancer, is by far much more common among the HIV population, as well as other which are less impact, like vagina, Hodgkin lymphoma, liver disease, and as you see here, and we are going to discuss this later, these in bold are all virus-related. So, why are we discussing this here? There's a very big complex association between HIV and aging and the effect they both have on the immune system, as we heard in the previous talk, and cancer risk factor, and we are going to describe a bit in the next few minutes about this issue. So yes, the HIV population is aging, as we discussed, and these are results from 2 different cohorts. The first picture is from the US, the second is from Denmark, and it's clear how there will be a huge increase comparing 2010 and 2030 in the aging population here, considering people more of 65 years old and people of more than 50 years old. So I'm not going to stop. We heard wonderful presentations and excellent explanation about what's happening with the immune system and aging. We heard about the inflammation, which is this chronic inflammation and activation of the immune system, and the immunosenescence, which this progressive decline of the immune response, reactivating chronic infections, as we heard of CMV, Epstein-Barr, that will, in its place, stimulate this both innate and adaptive response, and this will become a cycle. And this chronic inflammation will increase many of the aging-related diseases, as we already heard. So, but how does the immune system works with cancer? So there's a big burden from the immune system in cancer control. One thing is the possibility of eliminating many of the oncogenic viruses that we are going to see are very much related in cancer in HIV population. And on the other side, there's the cancer immune surveillance, where the immune system can detect and eliminate both pre-neoplastic as well as neoplastic cells. On the other side, when we have this immune activation going on, in some way there's a facilitating for cancer, because we are stimulating cell proliferation and production of procarcinogenic cytokines and growing factors. So back to HIV and cancer, we are going to discuss very little about the effects on the immune system, as we have already seen in other presentations. There are some possible direct effects of the HIV, mainly through the TAT and BPR proteins that probably plays a role with synergism with the oncogenic virus. They also produce impairment in the DNA repair, and we heard the impact that this may have in the possibility of developing cancer and alteration of the cell cycle. There's also a place for antiretrovirals, and also the high prevalence of bronchogenic factors that are present in the HIV population. So again, from HIV and the immune system, we have the immunodeficiency that will in turn impair the control of the oncogenic viruses. There's also this chronic immune activation, both in response to HIV as well as other viruses like CMV and Epstein-Barr. And there's also the response that we are already seeing from this bacterial intestinal translocation that will continue to trigger this immune activation and inflammation. And we also will have patterns the same as aging and immunosenescence in the HIV population, and many times even earlier in time, as one would expect from the non-HIV population, with changes in the T cell profile, higher CD8, lower CD4, and having defective cells, fibrosis of the lymphatic tissue that will in turn make it more complicated to recover the immune system. And all this will alter the possibility of controlling precancer and cancer cells, and actually we are here again facilitating cancer. So this, once again, a very simple schema of how all these factors interact and promote many of the aging-related diseases as we've seen, and in this case also cancer. And there are many biomarkers that have been studied, and we can see that many of these biomarkers are increased also in an untreated HIV infection, like this one here, but also when we treat HIV infection, these biomarkers keep on to be elevated, and that means that the inflammation is still there. And in many patients with HIV, with cancer diagnosis, many of these inflammatory biomarkers have been found to be also elevated, as well as some in HIV-negative controls. So, what about antiretrovirals and cancer? We can see this from two different points of view. On one side, if the use of combined antiretroviral therapy, as we see in one of the first graphs that I've shown, after the introduction of the combined treatment, there was a very steep decline, mainly for Kaposi sarcoma and non-Hodgkin lymphoma. What's happening with other non-AIDS defining cancers? It's a matter of controversy, and probably it will be changing a long time. Certainly, when people started to live longer, there was more time to develop these non-AIDS defining cancers, and they started to show up in a more frequent way. However, guidelines for treatment have been changing in the last 20 years a lot. So what about patients that spend a lot of time with CD4 below 200 or below 350 or below 500? Is the lower CD4 that matters? Is the time they spent with this low CD4 count, because we didn't know at that moment that we had to treat them all. And probably this will be something that will impact in what we are going to see in the future, because it's not the same someone that was diagnosed in the 80s or in the 90s and spent a lot of time with low CD4 that someone that is diagnosed right now and is starting treatment in the very few days before we have this CD4 depletion. On the other side, that's the effect of the antiretroviral therapies themselves. So there have been several reports from protease inhibitor, from zidovudine, for raltegravir, stating that they could be potentially oncogenic, but there was nothing clear about that. So far, we are not considering them oncogenic, but it's something that we may study. And on the other side, there's an anti-neoplastic effect of some drugs, for instance, the protease inhibitors in Kaposi sarcoma that are being under study. On top of that, we have a higher prevalence of the traditional cancer risk factors among HIV population. So as we see here, tobacco is associated with a lot of comorbidities and with a lot of cancer. And so as we can see here, the number of active tobacco users in this study doubled among people living with HIV, the one of the general population. And we have in this group about 20% of the cancers attributable to tobacco. And there's also a high use for alcohol. This was a study that stated that around 54% of people living with HIV would report any use, and about 25% would report heavy use of alcohol that one would expect to impact more in cancer. And then we have the oncogenic viruses, co-infection, which are really very frequent among HIV population. As we see here, there's around 40% of cancer affecting people living with HIV that are associated to viral infection, as opposed to around 4% in the general population. And as we see in this chart, these are just some, many of the cancers that are associated with virus and are very frequent among HIV population. We have cervical cancer here, anal cancer, with many of us are kind of, we don't like that definition about being AIDS defining or not non-AIDS defining. Actually, anal cancer now has a much bigger burden than cervical cancer among HIV population. Other genital cancer, we had non-Hodgkin disease related to Epstein-Barr virus and also in some forms KSHV virus. Hodgkin disease related also to Epstein-Barr, Kaposi sarcoma, and hepatocellular carcinoma, both from hepatitis C and hepatitis C. And as you see here, we have the 3 AIDS defining and many of the cancers that are having the highest burden among HIV population. And so far, lung cancer is the only cancer not related to a virus that is happening in excess in HIV population. And how do these viruses work? They alter the cell regulation, the cell cycle, they activate oncogenes, and they inhibit suppressor genes. For example, if we consider HPV of high risk, there are 2 proteins which are E6 and E7 that actually inhibits to attach and inhibits one of the 2 major cancer regulators genes, that's P53 and PRB. So just to give an example, we were talking about HPV. We know that people living with HIV has clearly a higher susceptibility. They have more incidence, more prevalence, more lesions, more reactivation and persistence of HPV infection. And this is clearly related to the impaired cellular immunity that they have. And it's really very frequent to find the co-infection. Around 75 to 95% of men who have sex with men living with HIV may have high-risk HPV, and around 40% of women living with HIV may have in their cervical region HPV too. So here we have just some of the cancers that are related to HPV. Cervical cancer was added in 1993 as part of the AIDS-defining conditions. It varies a lot, and it was the only AIDS-defining cancer that actually, when the combined antiretroviral therapy became available, it didn't show a clear decrease of its incidence. Some newer reports are talking about a decrease. However, there are studies that have shown that what's more important for this is having a screening program put in place. Anal cancer is becoming a very big problem. We have around 55 to 140 times higher frequency among people living with HIV, and this varies depending whether we are considering men who have sex with women, and the higher frequency it's among men who have sex with men. There are now a lot of screening practices put in place, and now we have to see exactly until we have the results of the anchor study how useful actually these practices are. And well, head and neck cancer, it's also a cancer that's more frequent among HIV population, and this has been also shown for other genital locations. What about HCV, which is another possible carcinogenic? It is estimated that around 25% of the people living with HIV are co-infected with HCV, and this number can even be higher, around 45% and even more in cohorts where there's a high burden of intravenous drug users. And now HCV is also becoming a problem among populations of men who have sex with men. And what's the impact of HIV among this infection? Well, we know that there's a reduced clearance of the infection, there are higher viral loads, there's an accelerated fibrosis, and this seems to be both from having more replication of HCV and also of some direct effect that the HIV may have. There's a higher incidence of descompensated cirrhosis, and as we see, there's an increased risk of hepatocarcinoma in about 4 times versus mono-infected patients. And the other thing is that the hepatocarcinoma usually develops in a much shorter time, about 10 years earlier than what one would expect in a mono-infected populations. We have new treatments. We have to see which will be the impact of this treatment in hepatocarcinoma in the long term. So, starting to close now, cancer in HIV populations, the things are changing. And as we see, the trends in cancer have been changing a long time, and about with combined antiretroviral therapy availability. And this is a study recently published from the NCI. And what is more interesting here is that when we compare 2010 to the estimates from 2030, there's a mild decrease in the total cancer burden, but most interestingly, for the first time, we are seeing non-infectious, non-AIDS-defining malignancies as the more frequent cancers in this population, which seems that will become lung cancer and prostate cancer. We need to be aware that, however, there's still an increased risk of Kaposi sarcoma even in this study, but it's the first time that we see this kind of shift. So what we can do to try to avoid cancer in this population? First, we need to test the patients. We see all the immunological reason for why this can be happening in HIV people. So the first things that we need to do is to test people. And then initiate combined antiretroviral therapy as soon as possible, because this in some way will keep the immune system of the patient as best as we can so far. Remember that any patient that is HIV positive and is diagnosed with cancers need to remain under treatment, adapted. There's another talk on polypharmacy. Maybe you need to touch something, but we need to keep this patient under treatment. Then, managing of oncogenic risk factors, trying to put the patients that are smoking on tobacco cessation programs and promote low use of alcohol. When appropriate, vaccinate the patients for HPV and HBV. We have now treatments both for HBV and HCV, and we need to see what's going on. And this, we are not going to discuss each topic, but just remember to screen the aging HIV population for cancer, and take in mind that here we are including not only some cancers that are more prevalent among the HIV population, such as anal, cervical, or hepatocellular carcinoma, but also we need to screen for those cancers that until now at least, are more prevalent in the general population. So just to wrap up, as we see, aging and HIV share a lot of immunological pathways that facilitate that these patients are more likely to have cancer. This on top, we had a lot of burden of other bronchogenic factors. Cancer is very important for HIV population, and things are changing, and we have to adapt our practice to this changing, and think about preventing and screening practices appropriate for this population. Thank you very much.
Okay, so I was thanking the speakers for their excellent talk and for keeping in time. So we have time for a few questions. Okay, so let's see. Yes, okay, Alan.
One of the questions that comes up, at least from the HIV perspective, is how much does underlying HIV, since we still have a reservoir of virus, and as we begin to look across the different comorbidities you've talked about today, you've talked about co-infections and cancer, but how much does HIV, its integration, cause or lead to some of the alterations in oncogenic sites and where it might integrate, since you're not eliminating it's an integrated provirus? Just wondering, as you're looking across muscle and the brain and even cancer, how do we look at HIV and how we're treating HIV, but still we're not curing HIV? So I'd like your thoughts about that.
Okay, thank you for your question. I'm more a clinician, but I think that that's an issue, and probably it's an issue in everything. My feeling when I was preparing this talk is that what I've said before, we have so many changes and so many challenges ahead with these different populations, and yes, we don't know. I mean, HIV seems to be part of the virus that are causing these pro-inflammatory states. So probably as long as we don't solve this issue, we are not be able to completely solve the rest of these consequences. So, I don't know.
Thank you for excellent presentation. I have a question for Dolores. Dolores, we as clinicians practicing infectious diseases, we see that our population is aging, okay? Some of them will get quote unquote normal Alzheimer or normal other degenerative diseases. But some of them will get HIV-associated cognitive disorders. Is there any way to differentiate if we are in front of one or the other?
Yes, in some cases. I mean, we can differentiate if we have fronto-subcortical cognitive impairment. For example, this is the more frequent spectrum of the cognitive decline associated with HIV is a fronto-subcortical cognitive impairment without cortical signs. However, for example, in Alzheimer's or in frontotemporal dementia, we can observe in neuropsychological tests other different patterns with more cortical affectation, for example, with language abnormalities, aphasia, with apraxia, and other things. So, yes, we can, in the neuropsychological test first, we can see the pattern, we can see if there are some cortical signs, which is frequent in neurodegenerative disease such as Alzheimer's or frontotemporal dementia or Lewy body dementia also. And then we can use other biomarkers, for example, like in HIV patients, like in other patients with HIV, for example, the PET, amyloid of PET, the PET glucose. We have other tests, ancillary tests that we can use to differentiate. So the answer is yes. Not always. In some cases we don't know, but we can try it.
Thank you so much for those enlightening presentations. We are really learning a lot. I actually work with women who are living with HIV, and we are having issues that are coming out for women who are aging, especially those who are above 50, women who are now in the menopausal stage. And it's like they are experiencing the same problems of painful feet. The whole left, right side, they have a pain from the waist going down. And some of them, from the clinics where we go, where we get our art, we have actually been scanned to see if there was something with our bone. And one lady was telling me last week that apparently her bone is actually destroyed on the waist and she's now limping. And we noticed that a lot of women who are on art, who are also menopausal, are having the same problem. So I just wanted to know whether there is any connection between HIV and aging for women who are on art and menopause. Maybe Valerie can answer that because I think it's more linked to your presentation. Thank you.
I don't know if you want to say something about sarcopenia.
Natural history related to menopause is very difficult so far because necessary to have more core. So far, in plenty of studies, aging person or person with more of 50 years, no aging person, are not allowed to participate in clinical trials, for example. The hormonal situation is really very important because it's implicated in multiple functions. So the decline of estrogen and other hormones is really very important. In fact, probably in muscle, some of the reasons implicated in the loss of type 2 fevers could be aggravated by menopause. So it's real and very interesting question, and it's necessary to discover and to find an explanation, because this story of the women with the menopause is necessary to discover.
Any other comments?
No, more or less the same. I mean, there would be a talk on bone afterwards, and I think that many of these aging, let's call it some way, issues that are happening, we know, for instance, with some drugs, there's been some relation with bone toxicity, so it might be a problem, we don't know. I think that at least I'm from Argentina, and the care of people living with HIV in Argentina are done by the ID specialist. And now the ID specialists have to learn a lot about internal medicine and other topics, because one thing is that now it's not just HIV, it's a lot of things that we have to take care. So I think this issue for women with menopausal care has also to be addressed, as it was said.
Well, you know, osteoporosis is one of the premature aging phenotypes associated with this inflammation and chronic inflammation. At menopause, women are more likely after menopause to undergo more bone loss. So this is amplified by all the pathogenesis related to HIV. So it can present itself at various type of clinical presentation, but not all of it is necessarily related to menopause, or to osteoporosis. So I think each individual case has to be taken into consideration. You mentioned in your talk the antiretrovirals and cancer, and you mentioned there might have been a protective effect of PIs on Kaposi sarcoma. Do you feel that it is a specific effect, or maybe just because of the control of the viral load and the restoration of the immune system?
Actually, it's something it's under study, and it's not clear. There haven't been no randomized controlled trials compare PIs to other drugs. But now that we are in the integrase inhibitors era, there has been some reports stating that when they turned from PIs to integrase inhibitors, the KS will come back. And then when they restore, but they are just reports, and nothing has so far been clearly defined.
If there is no other question, we're going to thank the speakers for their excellent talk, and we're going to meet for the photo op at the entrance.
speakers today. Pedro Khan will be the first speaker from Buenos Aires, and will be, let's say, leave the slides up, switching antiviral therapy. Then we will have a talk from Cécile Tremblay from the University of Montreal. She'll be reviewing the top 5 papers this past year on aging and HIV, and that'll be followed by a short session of the awardees for best submitted poster abstracts, and there'll be 4 awardees that will give short 5-minute talks following that. Thank you, and I'll just turn it over to Pedro, switching antiviral therapy to safer strategies based on integrase inhibitor. Pedro, thank you.
Thank you, Alan, and I would like to thank Eugenia and Bonaventura Clotet for organizing this wonderful meeting. And after the wonderful talks that we saw today, this one will be more clinical, and I will try to recap for you the current knowledge we have in regards to switching strategies. These are my disclosures. And first of all, there is a say that says, don't fix anything if it's not broken. So if a given antiretroviral regimen works, then you should not change it. Is this true? Well, it depends, because you may be aiming to different issues: improve adherence, maintain viral suppression and avoid resistance, reduce pill burden and dosing frequency, reduce tolerability and tolerability issues and toxicity issues, to avoid food or fluid requirements, as some antiretroviral drugs carry, to reduce drug-drug interactions, to adapt the regimen to pregnancy, and to adapt the regimen to aging patients, which is part of our main task during these 2 days. And last but not least, to reduce costs. And also, we may encounter in our patients concerning laboratory values, long-term toxicity, again, need to simplify, to adapt the regime to comorbidities, and in particular to adapt to some co-infections that are very, very frequent in some regions of the world, like hepatitis B and C and TB. So who should we switch? First of all, we need to ask ourselves how long has been our patient suppressed. And I will show you some examples of mistakes that we have done in some clinical trials in switching patients too early. Has the patient failed former antiretroviral regimes? So the history of antiretroviral therapy is a main issue, and it's pretty important. And who is asking for the switch? Is the patient asking for the switch? Does he come to your clinic and says, "Doctor, I'm fed up with so many pills, I want to change"? Or the patient is perfectly good, but you try to be, because you were at the last CROI or the last IAS conference and you saw a very nice switch study and said, let's move forward and switch. And maybe your patient doesn't want to do so. And sometimes you have an urgent need. For instance, Valeria was teaching us about cancer. Obviously, if you are starting chemotherapy, sometimes you need to switch to drugs that have a very low impact in terms of drug-drug interactions, like raltegravir or dolutegravir. So the principles, if we follow what the DHHS is telling us in the latest update, you can see that we need to review the antiretroviral history for possible virological failure. We need to review if we have available formal resistant tests in order to see if there's archived resistance. And in that particular case, be very careful before you start switching, because you can really ruin the current successful regime. When you switch from boosted PI to another class, if you don't know the full history about resistance, you need to be very careful in this regard. And eventually, if the case comes that you need to switch for any given reason and you're not sure, what you can do is consult an expert. This is very nice, because the guidelines are written by experts, and they tell us consult an expert. So they don't give us the real answer, no? And probably the exemption for this is when you switch inside the class. So you are using a PI and you switch to another PI and the patient is fully suppressed, then you shouldn't have any problem. You need to review the antiretroviral history for intolerance. Sometimes you switch back to a drug that the patient has been exposed to, and the patient doesn't like it because of the tolerability issues. The HLA, if you are considering abacavir, and again, drug-drug interactions. And as I told you, different issues that have been discussed, some of them, and they are very relevant for the aging population. Interesting, in this paper you can see that if your patient is switching because he had a side effect, then the patients who switch due to treatment-related side effect has statistically significant improvement in mean physical and mental health summary scores. But patients who switch for other reasons, they don't experience any improvement. So this is a kind of issue of ethics in switching. There is a wonderful paper published maybe 4 or 5 years ago by Tony Postniak in PLS Medicine, in which they were talking about the ethics of switching. That means if your patient doesn't need to switch, and there's no advantage for that, you have to think twice before you really proceed with switching. Now, this one. So let's learn from our mistakes. This is a typical case of failure, the switch-mark study, in which patients that were undetectable, at least for 3 months, as you can see here, they were randomized either to switch to raltegravir 400 milligrams BID and 2 nucleosides plus lopinavir ritonavir placebo, or patients continued in lopinavir ritonavir BID plus a placebo of raltegravir and 2 nucleosides. The primary endpoints were related to lipid concentrations and proportion of patients with less than 50 copies at week 24. So this worked very good in terms of lipids. As you can see here, triglycerides, non-HDL cholesterol, etc. What you see in blue is the advantage of the integrase inhibitor when compared to protease inhibitors. But when you go to viral suppression, even with nice numbers, 80% and 88% in switchmark 1 and switchmark 2 respectively, they couldn't show non-inferiority in regards to the comparison to lopinavir ritonavir. So this strategy seemed to be dead, but with a better designed study, as you can see here, did by the Spanish group of Esteban Martinez and co-workers, here the patient had at least 6 months of undetectable viral load, and they were checked in terms of formal resistance, and patients were randomized again, either to switch to raltegravir plus 2 nukes or to continue their current regimen with boosted PI and 2 nukes. And as you can see here, again, you see the advantages in terms of lipids, triglycerides, etc., total cholesterol, LDL, and also the efficacy was good in terms of maintaining viral suppression. So this is a very good example using exactly the same strategy, with the difference is that you select the appropriate patients, that you can have a failure or a success. Another more recent example is the use of the co-formulation of elbaitegravir and tenofovir FTC. And in this part of the slide, you see here the comparison with the ongoing boosted PI. There's non-inferior the strategy to switch to the integrase inhibitor. Here is in comparison to efavirenz. So both for PIs and for non-nucleosides, you can switch if you select the appropriate patients to, in this case, an elbaitegravir cobicistat-based regimen. No, I'm missing here. Okay. Then you have the striving study. In the striving study, co-formulated dolutegravir, abacavir, and 3-TC was compared to continuous antiretroviral therapy. Numbers are very similar, and non-inferiority was shown, and with a very high satisfaction score for patients that were switched to a single tablet regimen. This is a list of, even if you can read here, initially it's only for switching therapy. And as you can see here, the 2 integrase inhibitors that have been tested for this type of dual therapy strategies are dolutegravir, either with maraviroc, atazanavir ritonavir, darunavir ritonavir, rilpivirine, or 3-TC, or raltegravir with etravirine, 3-TC, darunavir ritonavir in 2 different studies. And I will show you some examples of those studies. One is the ASPIRE study. Again, patients that were undetectable for at least 6 months, without a history of resistance. And you can see here that the numbers are pretty similar. The proportion of patients that achieved less than 50 copies at the predefined time point. Then you have the French study by Christine Catlama using etravirine and raltegravir. 160 patients, and as you can see here in the graph, the virological success at week 48 was 99.4%. So patients that switched from their current regimens to this dual therapy with raltegravir and etravirine, almost 100% of them were okay. One patient failed, interesting enough. He selected the resistance to the non-nucleoside, but no resistance to raltegravir in this particular case. And the efficacy was maintained up to week 96. The flip side of this strategy is etravirine, which is a very expensive drug, at least in many parts of the world. So the issue of cost would not be an advantage in this particular case. And as you can see here, when you look to other parameters, in this case, they are very relevant for the aging population: GFR, cholesterol, HDL cholesterol, LDL cholesterol, non-HDL cholesterol, triglycerides, and the ratio between triglycerides and HDL. In all cases, there are significant advantages for the dual therapy regimen, the exception being the glucose levels, which didn't show any difference. So in terms of metabolic issues, this is a strategy that you could consider in order to reduce cardiovascular risk in your patients. Then we have the RALAM study, which is, again, a switch study in which a fixed dose combination of raltegravir and 3TC that was available in Europe. I don't know if it's still available here, but anyway, you can use those drugs in a two-pill regimen. Patients were randomized either to continue or to in a 2-to-1 ratio, or to switch to raltegravir 3TC. And as you can see here, when you go to the intention to treat analysis, 80% of the patients in the control arm and 96% in the raltegravir 3TC arm achieved less than 50 copies at week 24. Even if the others claim non-inferiority based on statistical management, I would say that the sample size is too small in order to drive conclusions. I would say it's a pilot study, it's encouraging, it's nice, but I would be very, very careful to talk about non-inferiority in this particular case. And as you can see here, the interesting is that this was not driven because of better tolerability. Because sometimes you can find that the regimen is non-inferior or even superior based on tolerability, but this is not the case. So the hazard ratio for in terms of adverse events was non-significantly different. Okay, so I think the main conclusion is that these results support the viability of a well-powered randomized trial. So the authors acknowledge that non-inferiority should not be claimed in this particular case. There is now a rollover trial with the new formulation of raltegravir, the QD formulation, and is in progress. And there's another trial that I could find in clinicaltrials.gov, which is the QD trial, in which patients are going to be switched to raltegravir QD. Need to be patients with non-detectable viral load in a stable regimen, one arm, 100 patients. Again, it will be a pilot study, will give us some information, not the definite one. Then we have well-powered studies like the SWORD-1 and SWORD-2. Remember that those were patients that were undetectable either on an integrase inhibitor-based regimen, non-nucleoside-based, or a PI-based, in all cases with 2 nukes. Patients were randomized to continue or to move to the dolutegravir plus rilpivirine, and the study will last until week 148, patients coming from different countries, and the primary endpoint is at week 48, proportion of patients in the intention to treat exposed snapshot FDA analysis with less than 50 copies. And you don't need to be a statistician to understand that the results are exactly the same, 95 to 95%, and the confidence interval, as you can see here, crosses 0, and it is below -4. So really, it's perfectly in consistency with the -4 + 4 limits for the 95% confidence interval that the FDA now requires for the switch study. So this particular study was the driver for the approval of the fixed dose combination of dolutegravir and rilpivirine that I learned that is already approved by the EMA here in Europe, not still launched in all the countries, at least not in Spain, I have been told. So, what about integrase inhibitors monotherapy? There was such an enthusiasm with dolutegravir 3TC. As you know, we have presented the results of the Gemini trial, dolutegravir plus 3TC compared to triple therapy based on dolutegravir recently at the wonderful Amsterdam conference. And some people said, well, okay, let's try dolutegravir in monotherapy. We saw some studies with good results, some studies with bad results. But the problem is the high rate of resistance selection in the integrase gene in the case of virological failure. And as you can see, there is a list of mutations that have been selected in different trials that you see the reference here in the bottom of the slide. So I think that we can now clear and loud say that dolutegravir monotherapy not only should not be used, but it should also not be tested furthermore as a strategy for patients that are undetectable, because the price that you pay by selecting this resistant mutation is extremely high. What about switch strategies for failing patients? Well, we have to go back in the old times, in the first study that showed that an integrase inhibitor could work, and this is the benchmark. It's nice to see that at that time, patients that were failing to nucleosides, non-nucleosides, and PIs, almost without hope, when dolutegravir came across, we could get a fantastic, almost 70% efficacy if you, in the optimized background regimen, you could select another drug on top of dolutegravir. So this was the first time in which we could see that the integrase inhibitors could work. Now we have, obviously, much more drugs. Another study that was basically done in developing countries, in which lopinavir ritonavir is the used to be until 2018 the preferred drug by WHO as a recommendation for patients failing in first line. They did a triple arm study in which they compared lopinavir ritonavir monotherapy in green, you can see that failed, and two strategies based on lopinavir ritonavir, one with raltegravir and the other one with 2 or 3 nucleosides. So you can read this study in two ways. You could say, okay, that's great, lopinavir ritonavir plus raltegravir is as good as lopinavir ritonavir plus 2 nukes. You could also say, why do I need raltegravir if I can do the job with 2 nukes? But anyway, it's another example that dual therapy could work in the context of failing patients. Then you have the Downing study that compared in a direct manner lopinavir ritonavir versus the dolutegravir in both cases with 2 nucleosides. And here it's not only non-inferior but also superior, as you can see, because the lower bound of the 95% confidence interval does not cross 0. So the superiority was shown in the Downing study that was also recently presented in Amsterdam. So what do the HHS guidelines tell us about switching? Well, you can switch within class, as I said, without a major problem. If you are switching between classes, again, if no drug resistance exists to new regimen components, you can do it safely. Switching to 2 drug regimens, so far the DHHS only recommends either ritonavir boosted PI plus 3TC, as it has been shown in several trials, like the OLE study and the GESIDA dual trial, both performed here in Spain. And it's quoted as a reasonable option when abacavir, TAF, or TDF are contraindicated or undesirable. Dolutegravir rilpivirine QD, based on the SWORD study that I showed you, and I assume that shortly dolutegravir 3TC will also be part of these recommendations. And then some supporting evidence for switching to 2 drug regimens, ritonavir boosted darunavir plus raltegravir, albeit the efficacy here, the DHHS says is only examined in treatment-naive patients, which is true in terms of randomized clinical trials, but there is an increasing amount of evidence in small trials with 100, 150 patients that show that this is probably a good strategy to pursue. Most important is what you should not do. You should not switch patients to boosted PI or integrase inhibitors monotherapy, particularly integrase inhibitor monotherapy, For boosted PIs, you may consider in some particular cases, but the question is, why not add 3TC and be on the safe side? Dolutegravir monotherapy, as I said, should not be used. We tested also, as other groups, the combination of atazanavir boosted and unboosted with raltegravir and didn't work. Maraviroc plus boosted PI didn't work either, and maraviroc plus raltegravir is also contraindicated. To finalize a couple of slides on new strategies in development, one is the long-acting new kid in the block of the integrase inhibitors. Cabotegravir is an injectable drug that is also available in an oral formulation. And as you can see here, they compared cabotegravir plus abacavir 3TC orally, and this was the regimen that patients received for the first 24 weeks. And after that, the patients were switched either to continue or switch to intramuscular cabotegravir and rilpivirine every 8 weeks or every 4 weeks. And as you can see here, the results, the best performance was for every 2 months injection. So we are in the verge of seeing a completely new era of long-acting treatments in which, based on the potency and the safety and good tolerability of this type of drugs, is opening really a new window in the field. Nevertheless, we need to see what our patients think, and this is a paper presented at CROI this year, in which patients were asked, would you move to a single pill taken once a week? Would you move to 2 shots given in the clinic every other month, as we have shown in the latest study, or 2 small plastic implants? You remember the implants that many ladies have for contraception? Well, I know at least that 3 companies are working on those type of implants that you could change every 6 months. As you can see, at least in this poll, the most popular form of new delivery of antiretroviral therapy is a single week taken once a week. So 2/3 of the population said that this would be the preferred switch, but more than 1/3 would go to the clinic every other month to get a shot. So I think that as long as we show more evidence and it becomes more popular, not for everybody, but for many patients, DOT, direct observed treatment, could be a good chance. And if we think about our aging population, which has, as we learned today, a lot of neurocognitive disorders. They may forget, they may not show up, they might be sick and not being able to come to the clinic, etc. It could be an interesting strategy for treating patients with dementia or with cognitive disorders. So, just to recap, remember that you can switch to improve adherence, to reduce the pill burden, to reduce tolerability and toxicity, to avoid food requirements, reduce drug-drug interactions, and to adapt the regimen to the different situations in which we see our patients, particularly aging patients, and again to reduce cost. And you can do so if your patient wants to switch or you have very compelling medical reasons to do so. It's nothing wrong to keep a patient 10 years with the same treatment if your patient is undetectable. So don't feel pushed that you need to switch because this is fashion and contemporary. Is the potency preserved? I think this is the most important question. When you are going to switch, is the new regimen as potent as the one that you are leaving aside? Again, take care of your patient previous failures and or resistance. Take at least 6 months to be on the safe side. And the longer the time your patient was undetectable, the safer the regimen will be. Have you considered potential drug interactions? And again, is your recommendation cost effective? If so, you can switch. So thank you very much for your attention. This photograph is for Eugenia Negrido, because she was at the Iguazu Falls recently.
Well, it's hard to follow Pedro. I'll try to do my best. So I was asked to pick 5 top publications from the last year. It's a difficult exercise to do in a field such as HIV and aging. So I said, what am I going to do? So I did a PubMed search. That's the first place to start, and looked only at published paper, not conference presentations, from September 2017 and September 2018. And then I figured, okay, so there are so many, how do I select? So I figured I would pick some on each class of scientific pillar and try to find the few paper that caught my interest. And I specifically excluded papers from topics that I knew would be covered by the other presenters today. So I knew there were presentations on microbiome, neurocognitive, and everything. So that's how I got to it. So let's start with the couple of papers in epidemiology related to HIV and aging. So this is the paper that was recently published on the new HIV diagnosis among adults aged 50 years or older in 31 European countries from 2014 to 2015 by Tavoshi and et al. And it's the EU EEA HIV surveillance network. So it's an observational analysis of surveillance data to identify trends. And the study population for this particular paper was all individuals aged 5 years or older who were newly diagnosed with HIV between January 1st, 2004 and December 31st, 2015, reported as part of the surveillance. And it was case-level anonymized data that were reported for each new diagnosis, and it includes demographic data and stage of disease and migration status. And older, please stay on your seat, is defined as 50 years or older. So that's what we have to live with. Okay. So of the 29,400 new cases that were reported in 2015, 5,017.3% were in older people at the table. So new HIV diagnosis among older people was 2.5 per 100,000 as opposed to 10.4 per 100,000, and the reported transmission route was significantly associated with age, with the higher proportion of cases attributed to heterosexual transmission in older people than in younger people. A higher proportion of older adults than of younger people were diagnosed late, with significantly lower median CD4 counts at diagnosis, and migration status was significantly associated with age, with older people were more likely to be native to the reporting country than younger people. So they did an analysis of which countries had seen increases in HIV diagnosis among older adults, and 16 countries. I have listed them here. And only in one country was there a significant decrease in the new cases in older adults, and that was Portugal. The average annual notification rate of new diagnosis among older adults was 4.4 per 100,000 men and 1.2 per 100,000 women, so more in men than women. And although the main route of transmission was heterosexual, the notification rate increased significantly in both sexes, from 3.5 to 4.8 in men and 1 to 1.2 in women. And in younger people, there was a significant increase in new diagnosis in men and boys, and a significant decrease in women and girls. So take-home messages from that study is that there is an increasing trend in new diagnosis among older adults in the European countries. There are clear geographical patterns. I didn't go into all the details, but there is ups and downs depending on what you're surveying that differs among the countries. Older adults are a group at potential risk for HIV, and older individuals are likely to be diagnosed late across the region. So, and the most probable reason are low self-perception of risk and misconceptions or scant knowledge about HIV that are frequently reported reasons for older adults not being tested. So this data points towards the compelling need to consider older adults as a relevant target for HIV prevention program, which is not something that we always think about. So that's the first paper I wanted to share with you. This is another epidemiologic paper. So it's a paper by DeConnick et al. And its non-AIDS mortality is higher among successfully treated people living with HIV compared with matched HIV negative control persons, a 15-year follow-up cohort study in Sweden. So it's ongoing debate whether the lifespan of successfully treated people living with HIV is comparable with that of the general population. The study was to compare all-cause mortality between HIV positive and HIV negative controlled person. There were 4,066 HIV positive matched against 8,702 negative controls according to age, sex, region of birth, and these are data from the Swedish National HIV Register and the Swedish Population Register. Their national HIV register is quite interesting because they capture everybody who's diagnosed with HIV from the onset and gather prospectively these data. So they covered a 15-year period. And to make a long story short, what they show is that after a 15-year follow-up period, successfully treated HIV positive people were found to be 3 times more likely to die when compared to HIV negative control. This is true at each stratum of follow-up that has been assessed. And a cause-specific hazard analysis was conducted to compare non-AIDS mortality rates between successfully treated HIV positive and HIV negative control. There were only 11 of 58 patients who died from an AIDS-related condition. And when you exclude AIDS-attributed mortality, still you see that there is an association of people, HIV-positive people are still 2.4 times more likely to die compared with HIV-negative person, and it was highly significant. And finally, the mortality by mode of transmission, mortality was higher among people who use drugs, but conducting a sensitivity analysis, stratifying for drug users, non-HIV positive, and there was still a 2-4 times more risk of dying than in HIV negative people. So even if you assess for the drug use, the risk was still there. So successfully treated HIV positive people, however, had the 76 reduced risk of mortality compared with untreated and unsuccessfully treated HIV patient over a 15-year period. That would come to sense, but it's sometimes it's good to have the data to show it. So, take-home messages from this study: the increased risk of mortality after 3 and 4 years in this study contrasts with other cohorts, where it has been seen that people living with HIV on ART with a baseline CD4 count above 350 had mortality rates comparable with the general population. It also contrasts with the COHERE study that followed HIV people on ART with CD4 count above 500 over a median of 3.5 years and found mortality rates similar to that of the general population. However, these other studies and also found that mortality was 4.2 times higher than the general population, with all people living with HIV were considered, and not only those who have achieved this high CD4 count. So this is important when we read these literature about life expectancy, that usually the conclusion are always when they consider, oh, if your viral load is suppressed and if you've reached CD4 count above a certain level. Well, this does not represent the majority of the people living with HIV. And then this is just a paper that was out like 2 days ago, and that Alan kindly showed me, and this morning I added those 2 slides. It is quite an interesting study, a meta-analysis of the global burden of atherosclerotic cardiovascular disease in people living with HIV. It should actually be in the next section. I kind of added the slides too quickly. So why is it so interesting? I didn't have time to review it in a lot of details, but it's the sample size. So they looked at 793,000 people living with HIV with a total follow-up of 3.5 million person years. So that's sample size. And the crude rate of cardiovascular disease was 61.8 per 10,000 person years. In comparison with individuals without HIV, the risk ratio for cardiovascular disease was 2.16. Over the past 26 years, the global population attributable fraction from cardiovascular disease attributable to HIV increased from 0.36% to 0.92%, and DALYs increased from 0.74% to 2.57%. So this is the forest plot that shows the different studies that were retained for their analysis, and that you can see that the ratio, the risk ratio looks around twofold increased risk. There was also marked regional variation, with most DALYs lost in sub-Saharan Africa, and the highest population attributable fraction and burden were observed in Swaziland, Botswana, and Lesotho. So I think it's good to take time to read this paper in much more detail, which I hope I'll have time to do later on, but I think it is a convincing evidence that there is a definite association with cardiovascular disease and HIV. So now we go to clinical science. So a couple of studies from Dr. Clotet and all of you have been involved with the EuroSIDA, aging and the evolution of comorbidities among HIV-positive individuals in the European cohort. So the goal is to describe the prevalence of comorbidities and risk factors. EuroSIDA, as you all know, is a multinational prospective cohort, collects data since 1994. Data is collected at 6 months interval. Epidemiological, demographic, C4 viral load are captured, and they compared two cross-sectional cohorts of HIV-positive adults under active follow-up, one in 2006 and then in 2014. Why they chose 2006, it was because it was at that time that they started collecting the creatinine data, and 2014 because they wanted to have current antiretroviral treatment. So 9,700 individuals were under active follow-up during 2006 and 12,000 during 2014, and 7,000 of those contributed to both cohorts. So the number of current smokers decreased from 51% to 44%. We're making some progress. There was the prevalence of risk factors and comorbidities generally increased in the older age group. There was a similar high prevalence of dyslipidemia, 70%, with the prevalence of diabetes increasing from 5.4 to 6.3%. In 2004, there was a small increase in the proportion of individuals who are overweight. Of course, this is Europe, it's not America. Things would be quite different. A large increase in the proportion of those with high blood pressure, 47 versus 60%, and the largest increase in prevalence was observed for chronic kidney disease, 4.1 to 6.9%, particularly in older individuals, where the proportion with kidney disease increased from 5.2 to 7.2 in the 50 to 60 year age group, and from 18 to 23% in those older than 60 years of age. And cardiovascular disease increased overall from 3.7 to 5%, with the highest prevalence in individual age 50 to 60 years and those above 60 years of age. So they did a univariate logistic regression modeling that showed that individual in the 2014 cohort had more than 2.5 higher odds of kidney disease compared to those in 2006. After adjusting for age group, the AOR for chronic kidney disease was 1.56. But after adjusting for other factors, including demographic, viral load, and CD4 count, HCV, smoking, etc., the year of the cohort was no longer associated with chronic kidney disease. So there's not a really increase over time. So older individual had significantly increased odds of kidney disease, and after adjustment, women were twice as likely to have kidney disease compared to men. This is something that should be explored. Those with lower CD4 cell count had higher odds of kidney disease with high viral load, and individuals with low body mass index at a small but not significant increase in the odds for kidney disease, while those who were overweight or obese had lower odds for kidney disease. After adjusting for age group, the odds of kidney disease were similar in 2006 and 2014. And finally, for cardiovascular disease, the 2014 cohort had higher odds of cardiovascular disease, 1.88, and after multivariable adjustment for age group, gender, region of Europe, mode of infection, viral load, antiretroviral therapy, etc., the year of the cohort was not associated with cardiovascular disease. Older individuals had higher odds for cardiovascular disease, and women were less likely to have cardiovascular disease. And current and past smokers had higher odds compared to those who had never smoked, and the odds for cardiovascular disease were also increased among those with hypertension and diabetes and kidney disease. So nothing surprising in all of these data, but it characterizes our epidemic, and it emphasizes the care that we need, the attention that we must pay to all of these factors. And I will just take you to the last line. As persons are aging and living longer, careful consideration should be given to clinical management of the older person living with HIV who face additional risk associated with HIV infection, immunosuppression, inflammation, as well as effects of ARVs, and need to avoid drug interactions and polypharmacy. So that was discussed a little bit this morning. So frailty, neurocognitive impairment, or both in predicting poor outcomes among adults living with human immunodeficiency virus. That was published in CID by Erlandson and colleagues. So this was an observational study of people living with HIV over 40 years of age who received randomized assignment of initial ART through the ACTG group and were followed prospectively in observational after their trial participation ended. It was 2013-2014. So they report data for the first 2 years. So they had questionnaires that went into quite a detail in terms of activities, daily activities, and falls, and frailty assessment was conducted. So of the 1,035 participants, 987 had both frailty and neurocognitive status assessed at baseline. So the data is based on these people. 4% were frail but didn't have any NCI, and 15% had NCI but no frailty, and 2% had both. And 79% had neither. So NCI was present in 11% of non-frail, 22% of pre-frail, and 32% of frail participants at baseline. So over 2 years of observation, 10% of participants experienced recurrent falls, 18% experienced worsening IADL limitations, and 2% died. 26% participants experienced at least one of these poor health outcomes. Poor health outcomes were most common among persons with both frailty and NCI, or with frailty without NCI. So anxiety, depression, medication changed the age-adjusted risk for the frailty without NCI exposure category by 10%, and therefore was excluded in the multivariable model. So in the models adjusted for anxiety and depression, the prevalence of a poor health outcome was 2.65 times greater among participants with both frailty and NCI compared to neither, and 2.26 times greater among participants with frailty but no NCI,
and 1.73 times greater among participants with no NCI but no frailty. So it seems that the factor that is the most associated with bad outcome, that drives most of it, is the frailty more than the NCI. So again, authors had hypothesized that the combination of both frailty and NCI would be stronger than either alone. This is partially supported by the data. The association observed for both frailty and NCI was stronger than NCI alone, but not stronger than for frailty alone. So needs to assess frailty in all of our patients. Okay. Next study was the cardiovascular disease and use of contemporary protease inhibitors, the DAD, International Prospective Multi-Cohort Study. It's always interesting to follow their findings over the years. We have learned a lot from this cohort, and this was related to the use of protease inhibitor and cardiovascular disease. So of all the participants assessed, 1,157 with cardiovascular disease. During a median follow-up of 6.96 years, 1,000 participants had a cardiovascular event at an incidence rate of 5.34 events per 1,000 person year of follow-up. The crude CVD incidence rate progressively increased from 4.91 events per 1,000 years in individual never exposed to ritonavir boosted darunavir to 13.6 events per 1,000 years in individual exposed for more than 6 years. For ritonavir boosted atazanavir, the incidence rates also increased, although less, from 5 events to 6.68 events. But when you adjusted for potential confounders in the primary model, where factors on the potential causal pathway from ritonavir-boosted PI used to CVD were fixed at baseline, only the darunavir association remained significantly associated with cardiovascular disease, adjusted IRR of 1.59. The adjusted IRR for atazanavir was not significant. After additional adjustment with time-updated values for the factors that could potentially be on the causal pathway to CVD, it did not affect the overall association, suggesting that the association between darunavir, ritonavir, and CVD is not mediated by any of these factors, and most notably not by dyslipidemia. So the number needed to treat to harm for ritonavir boosted darunavir stratified by the underlying estimated 5-year cardiovascular risk was 533 people for those at low risk of CVD and 15 people for those at high risk of CVD. So there were different effects between different protease inhibitors, with atazanavir not being associated after adjustment for the confounding factors, and darunavir boosted associated with cardiovascular disease, and and because you don't see it with all of the protease inhibitors, the authors claim that it is not a drug class effect, but it is more likely to be specific to some protease inhibitor. Okay, so these were the clinical. I'm going to take a couple of minutes. I'm going to skip a few of those on the basic science to just accelerate the time. So I just wanted to faire un clin d'œil to my friend Alan Landay and his very nice review on the microbiome and HIV. I will not go into the details because microbiome has been discussed in length this morning, but just to make you aware of that this review exists, very well written and very easy to follow, and that the findings that were discussed this morning, you can find all the references in this review paper. I'm gonna just accelerate. Okay, one thing that caught my attention is that there were a few papers on the TAT and its role in various aspects of premature aging in the last year. This one, it was in the scientific report, and it's related to the association of TAT with neurodegenerative phenotype with aging. So
in the central nervous system, HIV infects microglia and astroglia cells. However, production of viral particles in astrocytes is restricted. However, there is a chronic production of non-structural proteins, such as NEF-REV and TAT, in astrocyte that is thought to contribute to neuronal dysfunction. So this chronic low level of production of TAT has been proposed to contribute to neuronal damage over a prolonged period of time, but there was little evidence to support to this day. So they underwent to do a study in a transgenic mouse model that has a leaky tetracycline promoter system where they can play with the levels of TAT. And what they showed, to make a long story short, is that constitutive low-level TAT promoter activity is associated with structural modifications in the brain. Chronic low-level TAT expression is associated with synaptic change, and evidence for cytokine and cell stress in RTTA TAT mice were observed. And there was increased dentate gyrus volume that is associated with reactive gliosis. So they found a series of mechanisms by which TAT could create this, contribute to the neurodegeneration. During the same year, there are two other papers that I'm not going to discuss, but it has been shown that TAT alters neuronal intrinsic excitability. And there was another paper relating TAT to cardiovascular disease that I've listed here so that if you're interested, you may want to read. And finally, the last pillar of science, the social science. This is a Canadian paper. I thought I would at least put one Canadian paper in. It's unmet basic needs negatively affect health-related quality of life in people aging with HIV, and it is the results from the Positive Spaces, Healthy Places study. So they use a multiple linear regression model to examine the relationship between unmet basic needs and physical and mental quality of life by age strata. In a cross-sectional sample of 496 people living with HIV in Ontario, Canada. It was from this cohort that I just mentioned, and this is a 5-year observational cohort. 4 items on the 62-item questionnaire relating to food, clothing, housing, related costs, and risk of homelessness were analyzed. They particularly selected consciously a more vulnerable population out of their cohort to see if there were association. So an overwhelming majority of participants, 87%, reported unmet needs related to food, clothing, or housing. And the prevalence of unmet basic needs predicted substantially lower mean physical health and mental health summary scores in the 2 oldest group. So a majority of reported being unemployed, had been previously diagnosed with AIDS. The physical health summary score was low, and the health summary scores, mental health was depending on how you see it, was quite important. Role functioning scored the lowest, and the highest score was on the pain subscale. Significant difference among age categories with respect to gender, sexual orientation, and ethnicity. Gay or bisexual male participants were older than females, same-sex or heterosexual participants. So fewer individuals in the middle age and the old age groups reported unmet basic needs than did those in the youngest group, but it was not statistically significant, and it did not differ by gender, ethnic group, or sexual preference. So participants without a high school degree or those with a job at the time of the interview were more likely to report unmet basic needs than their peers. Among those with personal gross income below $1,000 a month, the 2 older groups were less likely than the youngest group to report unmet needs. Same for unemployed or those who had finished high school. So it was associated with poor physical health and poor mental health. And the middle-aged group, the older group, unemployed individual, Caucasian, and those with a previous diagnosis of AIDS and with a longer duration since HIV diagnosis, all had significantly lower physical health summary scores. being a female, unemployed, heterosexuals, low social support, and a personal income below $1,000 a month were all significantly associated with lower mental health summary score. So unmet needs lowered physical health scores to a greater degree in the oldest group than they did in the middle-aged group. So I chose the study because we need to take our patients globally in a holistic way. Unmet basic needs were significantly and negatively associated with lower physical health and mental health, even after controlling for potential confounders. There was very high prevalence of unmet needs. Age moderated the influence of unmet basic needs. Two older groups were well educated and enjoyed relatively high personal outcomes, so the association was not strong. The very high prevalence of unmet basic needs among people living with HIV have been magnified by the nature of the sample, which was purposefully recruited to represent a population with unstable housing. It did not differ by gender, race, or sexual orientation, which suggests that problems meeting basic needs is an across-the-board challenge for people living with HIV. So, you know, you think you're in the developed country, in Canada, and you still have 87% of the population who have trouble with food and with lodging and with these basic, so we're trying very hard to find cure and vaccine and everything, but food shouldn't be a bad idea. So these are the references for the study I presented. Thank you.
Can we get a round of applause for our two speakers before we call up our short abstract presenters? For Drs. Khan and Tremblay. Okay, we're now on to the fun part of the show. These are 5-minute quick presentations by our award winners here today. And I think Eugenia said that the check is in the mail for your award, so look for it sometime in the future. Anyways, the first speaker is Branca Pereira, who will be from the Chelsea Westminster, UK. So, Branca, you got 5 minutes, 3 slides.
I'll try.
I'll hold you to that.
Okay, I think it's this one. Good afternoon, everyone. My name is Branca. I work at Chelsea Westminster Hospital, and today I would like to describe our experience in managing cardiovascular risk in our cohort of patients living with HIV over the age of 50. So current guidelines recommend that cardiovascular risk assessment should be part of the routine management of people living with HIV. There are several risk assessment tools available. The UK guidelines recommend the use of Q-risk score to predict the 10-year risk of developing cardiovascular events, because this performed better than the Framingham score in the UK general population. However, both scores have been validated in the general population, so they may underestimate the risk in people living with HIV. Alternatively, HIV-specific cohorts, such as the one derived from the DAD study, may provide a better predictive value of cardiovascular risk in this population. However, this score has yet to be validated externally of the cohort. And then we also have coronary artery calcium CT scoring. So in recent years, it has been suggested that the CT scanning of the heart may provide a better predictive value of cardiovascular risk as compared to the previous screening tools. So basically, this is an imaging study that allows the detection of subclinical atherosclerosis. It detects the amount of calcified plaques in the 3 main vessels of the heart, and it provides with an absolute calcium score, as well as a centile, where the patient stands in terms of vascular age as compared to age and gender matched controls. So in our hospital, we have a dedicated clinic for all the patients over 50 years old, and we are lucky enough to offer this exam to all asymptomatic patients. And according to the centile where these patients are placed, if they're above the 5th centile, they should be offered preventive strategies. So if they're between the 5th and 75th centile, we offer him statin treatment for an aggressive lipid control, as well as lifestyle modifications, as well as consideration to change the antiretrovirus to a more cardioprotective regime. But if they're over the 75th centile, we refer them to cardiology. And now we've created this joint HIV cardiology clinic to allow a prompt assess and review of these patients, not only with a high CAC score, but also with other high-risk conditions of cardiovascular disease. So we performed a study, a retrospective analysis between May 2017 and April 2018, to analyze the concordance of the results between CACs and the traditional risk scores, Q-risk and Framingham. So during this period, 103 patients underwent a CT score, and 33 of these patients were 33 of these patients, 32% had abnormal results. So they were placed above the 5th centile. So you can see from the demographic table that the patients with higher scores, they are more likely to be older, as expected, more likely to be smokers, diabetic, slightly higher systolic blood pressure, and as expected, the cardiovascular risk calculated either by Framingham or Q-risk was higher in the patients with higher CAC scores. However, when we went to compare the correlation of CACs and Q-risk or Framingham in each specific patient, we found that among the 33 patients that had abnormal results, 14, highlighted in red, had a Q-risk score below 10. So they would have been placed in the low-risk group and would have been missed and offered the strategies to prevent cardiovascular risk. And the same was found with the Framingham score. So both scores actually performed really in a similar way in our cohort. So in conclusion, from our findings, what we want to highlight is the fact that traditional risk scores may indeed underestimate the risk, and they may fail to detect patients with subclinical atherosclerosis that may only be detected with CACs, and this supports the use of this screening tool in our clinical practice. Thank you very much.
Our next speaker is Anna Podubna, who is from the Ukraine. You've got 5 minutes.
Okay, thank you.
Thank you.
Good evening, dear colleagues. First, I would like to thank Ignazis for the invitation and for acceptance of our abstract. I have no conflict of interest to declare. The ongoing management of HIV aims to improve physical and psychological well-being and thus health-related quality of life. Also, very little is known about well-being of aged drug addicts living with HIV in Ukraine. So we aim to assess the health-related quality of life of people living with HIV who inject drugs and attend in regional cardiological clinic in Sumy, Ukraine, and healthy blood donors in order to identify the ways of its optimization and improvement. We conducted cross-sectional comparison parameters of well-being and defined 3 study populations: older group aged above 55 years, younger group, and population reference, healthy blood donors. All HIV-infected participants had no active manifestations of opportunistic infections and were retained in care. Concerned participants were interviewed to collect socio-demographical and behavior-related data using a study-specific case report form. Health-related quality of life was assessed using a specially developed questionnaire that includes combination of subjective and objective approach. The part of questionnaire for patient included close-ended questions to assess dimensions in physical, social, and spiritual component of health. The second part includes objective criteria from healthcare provider perspective with general health status, emotions, physical activity, ability to work, and presence of comorbidities. The 3 study populations were well matched except for the family status and educational level. Participants were mostly males with urban residence, and as we can see, a majority of HIV-infected participants of both age groups received antiretroviral therapy and opioid substitution therapy. The summary score of health-related quality of life for older group was lower compared to population reference and people who inject drugs from younger group. In general, 60% of aged drug addicts living with HIV, 85% of people who inject drugs in younger group, and 90% of donors were satisfied with their health status. Physical component of health was similar among all 3 study populations. There was no evidence for a difference in this index, while social component of health was, median score of social component of health was lower for aged people who inject drugs compared to donors and younger group, and the same situation for healthcare provider pattern. Spiritual component of health was lower for HIV-aged people who inject drugs living with HIV compared to population reference, but did not differ for HIV-infected participants of different age group. In conclusion, to our knowledge, this is the first report of quality of life in aged people who inject drugs living with HIV in Ukraine. The quality of life in this category of patients remains lower than in general population and younger group. And the low level of well-being in these patients is largely due to social and spiritual component of health, so an adequate access to social support and mental health care should become a key priority for this category of population. Of course, the most important limitation is the cross-sectional nature of the study. Also, these findings may not be generalizable to other studies with validated instruments or to non-injecting cohorts. Finally, we would like to express our deepest appreciation to our colleagues from Regional Oncological Clinic and our special thanks to all patients and donors for their participation on this study. Thank you very much for your attention.
Our next speaker is Liam Townsend. Liam comes from Trinity College in Dublin, Ireland. Liam?
All right. Thanks very much. So my name, as I said, is Liam Townsend. I'm an infectious disease physician currently working in St. James's Hospital in Dublin, and I'm also undertaking a PhD in the area of HIV and aging through Trinity College in Dublin as well. Firstly, I'd like to thank you for allowing me to present this case, but I have a confession, it's actually 2 cases, but they're a married couple, so it felt wrong to split them up just for the purposes of a presentation. So, I got the wrong one. I'll introduce you to them. So HIV first really became apparent in Dublin in the 80s, and it came along with an epidemic of heroin that mainly affected urban centres, specifically lower socioeconomic class, and particularly those living in high-density social housing, which is seen still in Dublin today. So they were married and they shared everything, and unfortunately that included needles, so both were diagnosed with HIV in 1985. And as was typical of HIV in the 80s, they were diagnosed late. So the husband presented with cerebral toxoplasmosis, and his wife presented in the same year with fungal tricuspid valve endocarditis. They did incredibly well with early treatment, both of their opportunistic infection and their virus itself. But as you can see, they've had a somewhat rocky course. As well as HIV, they were also co-infected with another bloodborne virus, hepatitis C. The wife, in this case, managed to clear it herself, but not before she became cirrhotic. Her husband also became cirrhotic and failed interferon and required DAAs to cure his hep C. He also suffered a spontaneous subdural hemorrhage, that's subsequent epilepsy. He has osteoporosis with a severe collis fracture of his wrist that still hasn't been aligned properly and has left him with severe disability. And he had a little bit of chest pain. It turns out he has marked stenosis of his LAD, so has had multiple stents put in. His wife has had a similarly rocky journey. Her ensuing valvulopathies following her endocarditis has meant she needed a pacemaker inserted. She has severe COPD, and she's recently started long-term oxygen therapy. And based on her MOCA cognitive assessments, she has mild cognitive impairment. So they've had a rough run despite doing very well. So where are we today? These are a couple aged 55 and 53. I've outlined already that they have multimorbidity. They have what goes hand in hand with multimorbidity, which is polypharmacy. So they're on double digits of tablets every day. And when you ask them what tablets they take, it's not the names, it's the blue one, the pink one, 2 whites in the evening. They're functionally impaired. As I've already said, the wife is oxygen dependent, she has mild cognitive impairment, and her husband, through a combination of osteoporosis and osteoarthritis, mobilizes with a walking stick. And I think, most importantly, they're socially excluded and have been all their lives. They've grown up in deprived circumstances. At the moment, they're living in a 1-bedroom flat or apartment. Also in that 1-bedroom flat and apartment is their son and their grandson. They've been off drugs for many years now. But I think it's clear that they're frail by any subjective measure, but are also frail objectively. Their frailty score is 4 in both cases, which marks them out as frail. And on the Rockwood score, they would score mild to moderate in frailty. And I think this is leading us to a paradigm shift as HIV physicians. It's well established, and this is some really nice data from Schoten et al., it was published in CID in 2014, that shows people with HIV suffer non-AIDS associated multimorbidity at a much earlier age than their HIV negative matched. And that's been echoed throughout today. But most concerning is that they're suffering these multimorbidities in ages that wouldn't traditionally be considered geriatric populations. So I think the paradigm shift that we need to do is shift away from a chronological view of age, where you look at someone's date of birth and treat them based on that, and look at a functional or biological calculation of age. Because at the moment, this patient is falling down that socially excluded gap because there are no services to provide functional support for independent living for people until they reach the geriatric cohort age, which tends to be over 65. There is, of course, the ability for them to go into a nursing home, but I think from all they've come through to tell them, I'm sorry, we can't help you live alone anymore, you need to go into a nursing home at 55, I think that's a failure of our system. So I think as HIV physicians and researchers, we are going to see a growing population of people who have multimorbidity, are frail, but aren't traditionally viewed as geriatric, and we are in an ideal position to advocate and give them a voice, given that they are so often marginalised. And I think that case hopefully illustrates that point. So all that remains for me now is to thank you very much for your attention. Thank you.
Our next speaker will be Katerina Yurko, who's also from the Ukraine. Katerina.
Dear colleagues, now I would like to talk with you about clinical case from my practice. So the patient, 58 years old, It's not first slide. Thank you. At December of 2017, the patient complained of general weakness in right leg and arm, tremor of hands and head, disorders and difficulties of speech, scanning speech, nausea, severe headache, and decreased memory and vision. During the hospitalization on neurological department, this patient, general condition of this patient was severe due to neurological symptoms, such as tremor of hands and head, motor and sensory dysphasia, asymmetry of nasolabial folds, right-sided hemiparesis, nystagmus, and many other neurological symptoms, ataxia in the Romberg pose. Blood pressure was high, 170 to 90, and the preliminary diagnosis was made a stroke. But on examination, all groups of lymph nodes was enlarged. In CBC, anemia, leukopenia, and thrombocytopenia was detected. HIV RNA by polymerase chain reaction in blood was detected. The amount of CD4 T lymphocytes was very low, 47 cells per microliter. The ELISA test showed positive antibodies, very high level of antibodies IgG to Toxoplasma gondii. MRI of the brain, we can see in this picture multiple zones of pathological intensivity in both cerebral hemispheres, surrounded by perifocal edema. And from anamnesis of life, it is known that more than 10 years ago, she had unprotected sexual contact. That's why the final diagnosis, HIV infection, fourth clinical stage, cerebral toxoplasmosis. The therapy of this patient was prescribed clindamycin 600 milligram 4 times daily, and trimethoprim sulfamethoxazole 960 milligram 3 times per day. Against 45 days of treatment, this patient noted the positive dynamic, for example, improvement of general condition, disappeared severe headache, and of course we can see a regressed phenomena of gemiparesis, improved memory and vision. And during the MRI, we can see positive dynamic. That's why during same clinical symptoms and high percentage of late diagnosis of HIV in Ukraine, of course, for elder patient, we should perform differential diagnosis between neurological diseases and toxoplasmosis, and on the background of HIV infection. And conclusion, considering the fact that combination of toxoplasmosis in HIV positive elderly patient is not frequent pathology in doctor practice. In my opinion, all doctors should remember and be able to distinguish this pathology from these other neurological diseases and to prevent development of severe complication. Thank you very much for your attention.
We need 2 chairs. Oh, here, where's, oh, okay. All right, do we have any questions from the audience for any of the presenters today? Again, I think this was a great interactive discussion, and maybe Pedro, you could comment on, from as a senior clinician in the field, from what you heard from some of the case presentations, and the idea of, I think Liam brought it up, is that services provided for the older for the patients at age 50 who aren't really that old are not in the, for what we would do, at least in the US, in a gerontology clinic, where the starting age is 65 or 70, where you start to see. So how is that care taken in Argentina, and how do you see the field in general if we're trying to move to caring for our aging HIV population, which are really in their 50s and 55, and we heard from our case presentations today, that's where we're seeing some of the early non-AIDS comorbidities.
Yes, thank you. As a first comment, I think we need to be reminded that the problem of the aging population is only growing, and as time goes by, we will see more and more people living with HIV in their 50s, in their 60s, in their 70s. And this is due to 2 different issues. One is that our patients that used to be 35 years old 20 years ago are now 55 because they are living longer, but also that people are acquiring HIV after 50, after they become 50. I think it was Woody Allen that said that the most perfect institution that the human being has created was marriage, but it was created at a time in which people died at their 50s. And now that people live longer, people tend to split and start again new relationship. They are engaged in dangerous or risky sexual behavior because they don't have the perception that they could be in some way at risk. And when they complain about any kind of symptoms, they go to the clinician, and the clinician would never say, "Oh, this gray hair man could have HIV." So that's the reason why Cecile very wisely showed us that older patients are also amongst those that are diagnosed later in the disease. Having said that, the problem we have in our region in general, not only in Argentina, is that our HIV clinics are overwhelmed with patients. And the issue is that I would say a fair proportion of HIV clinicians or HIV treaters, they don't want to deal with other diseases. They have enough with, taking care of viral load, CD4s, and etc. On the other hand, many clinicians don't want to deal with HIV. So I think that the way out probably is to try to more and more work in the direction of what the Americans call a one-stop shop. I mean, patient cannot come to the HIV physician and then go to the diabetologist and then go to the osteoarticular doctor and to the cancer doctor and so on. So we need to integrate our clinics in a kind of horizontal way. It's easy to say, it's difficult to do, but I think that if we are really aiming to the 90-90-90 goals, even if we are falling short, but we are aiming to, and we try to detect more people living with HIV, we will have more work in our clinics, and we need really to strengthen the system by integrating internal medicine tools with infectious diseases. In some countries, to be an infectious disease physician, you need to have a background on internal medicine. It's not the case in all countries. In some countries, with 1 year of internal medicine, you can get a 3-year or 4 years board certification in infectious diseases or infectious diseases, and this makes a big difference for people treating these other diseases. So I am obviously much more in favor of having infectious diseases trainees after having done a 3 or 4 year residency in internal medicine, but this is not always the case, so it's a difficult point.
Branka, maybe you could comment, how did Chelsea Westminster go to creating an HIV cardiology clinic? I'm curious how that came about.
So the idea of this clinic, thank you, is exactly to follow on the one-stop shop or the one-stop clinic, to allow patients to come to the place where they used to come for the HIV clinic and have a specialized cardiology assessment in the right time. So we don't have to refer them and wait sometimes longer than the patients should. And so far, the clinic has been very popular, and it's difficult to see all the patients that should be seen in the right time, but we're trying our best, and it's so far been a very good experience. Yeah.
Great. Cecile, would you like to comment on from the how you might handle this in Canada? We're going to each of our countries will solve the world's problems today.
Well, Canada is very large, and practices are very different from one end to the other. So even in the care of HIV, the type of clinics that are set up in our institution, so we're the hospital, so we have a clinic dedicated to HIV, and it's mostly infectious disease people who take care of HIV patients. And in our case, we used to have the training of only 2 years of internal medicine, and the rest is infectious disease and medical micro, so we have both. So we have lots of years of ID and medical micro, not a lot of internal medicine. But we also have a model where we have family doctors who are primary care physicians for HIV people, who will easily do and think about the screening for, what you're supposed to do for prevention and screening at all the steps, more than we may. Over the years, we have become used to it, and we've learned through all of this, what we've learned, unfortunately, with the premature aging phenotypes, it reminds us of what we need to do. But now the program has changed in Quebec, and now it is the regular 3 years of internal medicine and then infectious disease and medical micro. So we have like a 3-leg program, and I think it's going to help having this notion of taking in charge of the entire person, not just the virus or the microbe.
Are there other comments from the audience? Peter, did you want to say something?
Is it working? Yes. I totally agree, and I think actually the medical curricula should reflect that, because we've had people trained in internal medicine who then subspecialized in different areas, including HIV, but also others. And I think this issue of seeing elderly or older patients happens in other fields as well. And I think our colleagues in other fields suffer from the exact same problem. So I think what I tell the fellows is to make sure that they maintain their general internal medicine knowledge in order to deal with these problems, and I think it has to happen across the board.
Yeah, and then Pedro, and then.
I think that the way forward is to think about the modern therapy we are using now. In the old days, in which you had to deal with DDI, D4T, Indinavir, etc., it was really complicated for physicians to deal with. But nowadays, if we are aiming to test and treat, and we are using drugs that is a single tablet regimen, or in the worst case scenario, two pills, almost no side effects, in patients that are otherwise healthy, this should be general practitioners taking care about this, and we as infectious diseases should take care of the complicated cases with the resistance, opportunistic infections, etc. Because there is no way out to really get to the 90-90-90 if we keep in a kind of silo the treatment of people living with HIV.
I would like to share some information about my country. Ukraine has a strong focus on delivery and support for people living with HIV, and especially for aged people living with HIV. But now we have to face a lot of challenges, and the first point is limited resources and adequate access to these resources. For instance, tests for CD4 cells count, not always available, viral load, not always available. And the second point is non-integrated services. For example, if we have a patient and this patient is sick and we admitted him to infectious disease hospital, in case of TB, we transfer this patient to anti-TB hospital. After discharging, he continues to receive anti-TB treatment in another institution, outpatient clinic. If this patient, drug user, he receives opioid substitution therapy in other institution, oncological clinic, and at last he receives antiretrovirals in HIV outpatient clinic, and it's also a huge problem for us.
Any other final comments or thoughts from anybody in the audience? Yes.
So, I'll give you a perspective from Africa. I come from Kenya, work as a specialist in internal medicine. So for Africa, our epidemic became a public health priority. So when we rolled out ART treatment, it meant the nurses, the physician assistants were the main people treating people with HIV so that we could get everybody on treatment. That meant the internal medicine specialists, the doctors, kind of took a backseat to HIV treatment, unfortunately, because they were like, this is the thing that's funded by other partners and it will be rolled along. But now we're seeing patients getting older, living longer. Diabetes, hypertension, chronic kidney disease, strokes, you name it, are affecting our patients. And now is when the internal medicine physicians are now coming back on board and trying to find their role in the HIV world in my country. So it's always a challenge, but we try to keep involved, especially in patients who are failing, patients on multiple drugs. I feel for Liam's case, because I've had similar cases where someone is on multiple drugs and you're trying to figure out how to manage the interactions and everything. So it's always a big challenge.
Well, thank you. Again, we'll conclude this if there's no other final comments, and thank all the speakers and presenters. I'm going to ask Naomi Shegloff to come up. Naomi will tell us what we're going to be doing next in terms of the presentation and our dinner for the evening. So everybody can see it. Oh, I'm sorry. Sorry. Okay. Thank you.
Hi, everybody. I know it's been a full and long day, so bear with me for just a minute. My name's Naomi Shegloff. This is my collaborator, Katja Heinemann. Together, we're the co-directors of The Graying of AIDS. Among other things, you found a little book in your tote bag. That is a sample of some of the portraits and some interview quotes that come from our project Stories from an Aging Pandemic. 2012, 2014, and 2016, all three of those international AIDS conferences, we created a participatory installation where Katja, who's a photographer and photojournalist, created a pop-up photo studio, and I did brief oral history interviews with people. Some of the questions were uniform, everybody got the same questions, but some of the questions we collected from anybody who wanted to submit them. When you were having coffee and cookies earlier, you saw some of the questions that people wrote. Some people don't feel comfortable having their name, their face, and their story on the internet. Ultimately, all of the photos and edited versions of the interviews wind up in an internet archive. So you'll see in some places, people who didn't want to be photographed and interviewed just wrote their answers on those pieces of paper. In a minute, we're going to go downstairs. You're going to have dinner, which I'm sure you're thrilled about. But you'll also see a projected exhibition of some additional portraits, some of the same ones in your book, but also more portraits and brief interview excerpts. We invite you to check it out, but also, if you have some time, there are longer versions of interviews available on our web archive. We're in the process of tagging them to make them more searchable, so we're not quite there yet, but hopefully you'll find them useful. I really appreciate coming from more of a health educator background, being exposed to some of the science, and both on the clinical and laboratory science. But one of the things I also really want to invite you to include is the voices of people who are actually living with the virus, age 50 and older. There are one person who we had the pleasure of working with in Durban, Tendai, is here today. And you'll see her in the slideshow as well. But please, every time you hear about these studies, don't forget that these are people who have their own perspective on things as well. And I invite you to meet some of them downstairs while you enjoy dinner. We're going to head down there now. Thanks.